Welcome to our second day here at Kreatikon and the first session for today. Ole Hösja will tell us more about baraminology. And if you're here sitting and thinking, I'm not really sure what this topic is about, you are not alone. I think I can learn something new here today. And this session will go until 11, and then we will have a short coffee break. And before we start, I will begin with a prayer and ask God for his blessings for this event. Thank you, Jesus, for this new day and for the opportunity to hear more about your greatness. And please bless this day and this session here from Ola. Amen. And now I will hand over to you, Ola.
So, thanks a lot. And thanks a lot for the invitation to come here to Kreatikon 25. It's a wonderful conference with so many attendances, and so I'm so inspired. So many young people taking part in this conference. And my name is Ola Hösja. I live in Sweden, a suburb of Stockholm. I'm 60 years old. I have 2 daughters and 1 son-in-law, and I'm a member of a Stockholm Vineyard Church in Stockholm. And I work as a university professor in mathematical statistics at Stockholm University. But I'm also part of the Swedish creationist organization Genesis, and I want to send my warmest greetings from them. And the topic of this talk today is an overview of baraminology, the systematic scientific study of biblical kinds. So we read in the first chapter of Genesis that God created animals and plants after their kinds. And it turns out that this is a very reliable description, and science can confirm this. And what I'm going to say today, this talk is based on one chapter from this book. Last year in October.
You don't need this microphone.
Oh yes, you're right. Yeah, thank you. Last year we had the first Nordic research creation, pedagogical illustrations and explanations of the fact that God created animals and plants after their life. And that was really necessary for all the animals to, if that would not have been the case, there would have been many more species 4,500 years ago, but they could all be harbored into the ark. So that was a short introduction. So let me give an outline of this talk. So I will start mention about the bushes of life model, and that is really another name for God creating animals and plants after their kinds. And then I will turn on and give a brief history of the systematic study of created kinds, and that's where the word baraminology comes from. It's a Hebrew word. I will come back to that. And then you could say that this systematic study has 3 major goals. And the first goal is to really find those kinds. Which are the created kinds? Which groups of animals do they correspond to today? And then the second goal is to find the mechanisms of divergence and speciation within kinds. So for each group, each kind, it started with a single pair or a first generation, but today we have many species within each kind, and it's like a bush. But the different bushes, the different kinds, they don't have common ancestry. So this speciation or adaptation to environmental changes, that is not evolution, because it doesn't create new structures. And then we have the third goal of baraminology. Given that you found those created kinds, trying to find the ancestral history of each kind. And then we will zoom in to humans and ask the question, is it a kind of its own? So do we humans have unique ancestry from Adam and Eve that were created uniquely in the image of God without ancestry with animals, as the Bible tells? And then I will wrap up with some conclusions. So let us start with the bushes of life model. So you could roughly talk about 3 models for the diversity of life. The classical creationist old model is a species fixity model, that God created species ready 6,000 years ago, and then they haven't really changed. And then we have the bushes of life model, which I will talk about today. It's also creationist, but it says that God created the first pair within each kind, and then there has been speciation within each kind, but the different bushes, the different kinds, they don't have common ancestry. And then we have the evolutionary tree of life model, where all living species today, they all descend from first unicellular organisms. So they all have a common ancestry, and that gives us the tree of life model, usually with a much longer time frame. Instead of 6,000 years ago, several billion years. And you could say that the bushes of life model is in between the species of fixity and tree of life model. Although the bushes of life model is creationist, it does allow for speciation. But as I said, it's another type of speciation that evolutionary biology holds, because evolutionary biology, the tree of life model, requires that that new species with completely new types of organs, wings and eyes and echolocation systems and so on, evolve. So let me give some examples of kinds and bushes. So although strictly biblically humans are not a kind, because we are created uniquely of our own, but we can also think of humans as our own kind. So humans, modern humans, Homo sapiens, but then also include Neanderthals, Denisovans, Homo erectus, then they all have a common ancestry from Adam and Eve. So we can say that they form their own kinds. But then we also have great apes, chimpanzees, gorillas, orangutans, they form their own kind, Pongidae. And then we have the dog kind, Canidae, consisting of wolves, foxes, breeding dogs, and so on. We have the cat kind, Felidae, including tigers, lions, jaguars, cats, and so on. And so these are 4 examples of kinds. But let us now go back to the Bible and ask ourselves, is there a biblical motivation for kinds in general? And first of all, as I mentioned earlier, we go to the first chapter of the Bible, Genesis chapter 1, and in several verses, we are told that plants and animals were created after their kind. And then at the end of the first chapter, and then the creation story continues in the second chapter of Genesis, telling us that humans were created uniquely. So we can think of humans as its own kind. But then we have diversification, we have speciation within each kind. And we can also think of a biblical motivation for that, because not only humans, but also animals were commanded to multiply and fill the earth. And we can think of God designing mechanisms of environmental adaptation, including speciation of animals within kinds. And if I'm allowed to speculate a little bit, we know that God can look into the future. He knew about the fall of man, and he knew that also the nature will start to decay after the fall of man, and that would create some harsh environmental conditions. So maybe God created these mechanisms of adaptation also having those harsh environmental changes in mind from early on. But then, of course, we have a limiting factor, the flood of Noah, 1,500 years later, 4,500 years later, which also relates to the title of this German book that I mentioned about. You could phrase this as a bottleneck. So the size of each animal kind was reduced to 2 or 14. So in some way they had to restart those kinds after the flood of Noah. So that was a biblical motivation for the Bushes of Life model. And let's now relate this to baraminology, which is the title of this talk. And baramin is a Hebrew word for created kind. It's really composed of 2 Hebrew words put together. And baraminology is a systematic study or systematic scientific study of created kind. And the word was introduced by the creationist Frank Marsh in 1944. And as I mentioned, when I gave the outline of this talk, there are three major goals of baraminology. First of all, to identify all created kinds, which are they? And then find which are the mechanisms that give this speciation and divergence within each created kind. And then thirdly, given that we have found the created kind, find their ancestral history. Which individuals or which species within each kind are more closely related to each other. And in order to study this, we will actually be helped by taxonomy. And both secular sciences that hold on to the Tree of Life model and creationists are helped by taxonomy, because in taxonomy you sort life hierarchically into levels. We have species at the next level, the genus, at the next hierarchical level, higher up, family. We have the order, we have the class, phyla, kingdom, and domain. And an evolutionary biologist think that all species are related through the Tree of Life, but as a creationist, you believe that species are only related within the same bush. But we can still have the taxonomic levels can still help you, because the lower orders of taxonomy can correspond to species within a bush. And then, of course, we can have common design between bushes, so that some bushes can be more related than others, because God, as a designer, can reuse the same principles for different bushes. And that leads us to the question, which is the typical taxonomic level of a bush kind? Is it at the species level, the genus level, the family level, or the order level? And Eric Wassmann, more than 100 years ago, a creationist, he said that, well, it actually depends on the study group. And 15 years later, George McCready Price, another creationist, he said that, yes, but family is often a good candidate for being the right taxonomic level for a biblical kind. And it turns out that both of these predictions were good. So let's suggest some possible kind levels. And I will partly go back to the examples I gave earlier of biblical kinds. We have humans, as I said, including modern humans, Neanderthals, Denisovans, and we think of all of them as descendants of Adam and Eve. And then humans form its own kind at the species level. On the other hand, the other 3 suggested kinds that I mentioned, great apes, the dog kind, and the cat kind, taxonomically they are all defined on the family level. Whereas many creationist researchers think that chicken-like birds, galliforms, they form its own kind, and it's yet another hierarchical level higher up at the order level. So the hierarchical level of a kind might depend on the study group. So let's now turn to the history of the Bushes of Life model and its alternatives. And up till the 18th century, young Earth creationism was very mainstream, so most Christians were creationists, and the species fixity model dominated. But my countryman, the Swedish botanist Carl Linnaeus, the father of taxonomy, he also advocated species fixity. But at the end of his life, he actually changed his mind and allowed for some speciation within, actually. But he was still a creationist. So that means that already in the 18th century, he opened up for the bushes of life model. But then we know that in at the end of the 18th century and in the early 19th century, many scientists adopted high ages after the work of geologists, the Scottish geologists James Hutton and Charles Lyell. And that in turn gave rise to tree of life models because you needed long time frames for evolutionary model. The French scientist Lamarck, he proposed an evolutionary theory in the early 19th century, and then about at the same time Alfred Wallace and Charles Darwin in the late 1850s. But even at this time there were many scholars who criticized the tree of life model, and they favored various versions of the bushes of life model. For instance, the British botanist William Herbert in the 1820s, and also the paleontologist Richard Owen in the 1850s, and he was a critic of Darwin's theory. But in spite of this, that there were many critics of evolutionary theory who favored some versions of the bushes of life model, in his famous book, The Origin of Species, Charles Darwin, he pretty much equated creationism with species fixity. And there were so many signs of speciation taking place. So that sort of gave a negative flavor of the creationist camp. So let's continue now to the 20th century. And then we know that the tree of life model, it gained wide acceptance in the 1940s, because then evolutionary theory was married with genetics and giving genetic explanations for the occurrence of new species through macroevolution. And that's called the modern synthesis or neo-Darwinisms. And instrumental persons in this process were the geologist George Gaylord Simpson, the biologist Julian Huxley and Ernst Mayr, and the geneticist Theodosius Dobzhansky. But there were still many creationists around during the first half of the 20th century, and increasingly many of them favored the bushes of life model rather than the species fixity model. For instance, Eric Wassmann, that I mentioned about, Harold Morton, George Macready Price, that I mentioned about, Dudley Whitney, Harold Clark, and Byron Nelson. And then, as I mentioned, in the 1940s, Frank Marsh, he coined the term baraminology for the systematic study of created kinds. And I would say that from the 1950s, species fixity became a minority position among creationists in favor of the bushes of life model, because there were so many examples of speciation taking place. So that was some brief history of the bushes of life models and alternatives. So let's go back now to the three major goals of baraminology. First of all, to identify all created kinds. Which are they? Secondly, find mechanisms of divergence and speciations within each kind. How does it work when new species form within each kind? And thirdly, to find the ancestral history of each created kind. Which species within each kind are more closely related than others? So let's turn now to the first goal of baraminology, to identify the created kinds. And there are actually three main methods for doing that. The first one is so-called hybridization, or looking at hybridization observations or experiments, for which species within a kind is it possible for them to have offspring, and that signifies that they belong to the same kind. The cognitive method, that we can use human senses to help us identifying biblical kinds, and then something called statistical baraminology, where you use statistical methods for very large data sets in order to group species into kinds. Let's start with hybridizations, and suppose that two organisms or individuals that they can hybridize and have offspring. And reproduction, that's an extremely complicated process. Here we have an example of embryological development from the first fertilized cell. In order for this embryological development to take place, the two organisms, they must be related in order to have viable offspring. For this reason, Frank Marsh in 1944, he said that, yes, hybridization is a sufficient condition for a pair to belong to the same created kind. It's not a necessary condition, because a pair of species, even though they have common ancestry within their bush, they can lose the ability to have offspring. But if they belong to the same species, Leitner et al. In an article from 2011, they said that, yes, then more is required, because then the pair also needs to have fertile offspring. We have, for instance, donkeys and horses. They can have offspring, mules, but they are not fertile, but they still belong to the same kind. So they hybridize, but they don't have fertile offspring, because they don't belong to the same species. So can we define hybridization within kinds more accurately? Well, as I said, in order for the pair to belong to the same kind, the offspring doesn't need to be fertile. And it doesn't even need to survive embryological development and be born alive, but it needs to pass a certain stage of this embryological development. And that was suggested by Siegfried Scherer, your countryman, in 1993. And then you could use hybridization data between different pairs of species in order to build up different groups and thereby suggest different created kinds. So let's look more closely into the cat family and hybridization within the cat family. So as I mentioned, the cat family, it's a taxonomically it's a family. It consists of 40 species and it has 14 genera. So genus is a level between species and family. And here we have hybridization data for 6 genera. We see they are notated as G1, G2, and so forth. So we have different species, lion, tiger, leopard, and so on. And if there is a successful hybridization, it's marked with a cross. So we see that several of these different species, they can hybridize. And probably this data is incomplete, but even for this incomplete data, we can see that we have 2 hybridization chains. So at least we have 2 groups within the cat family: lion, tigers, leopard, pumas, and ocelots forming one group, and the other part, the other group. But it's still generally believed that this whole group forms its own kind. So that's the first method for identifying kinds, hybridization. The second method is the so-called cognitum method. So, and the general idea, going back from a paper by Saunders and Wise from 2003, is that it is possible to use human senses and sort species into groups. And those groups need not be at the kind level, because we can think of, yes, all the mammals, which taxonomically is higher up, they have certain shared features, or we can also look at species within a kind, like all foxes, they share certain features, and we can use human senses in order to do that grouping. And in doing so, that it's a useful complement to hybridization. If the hybridization experiments are not really complete, we can use human senses to sort of suggest that, yes, this is really a biblical kind. And let us give a biblical motivation for using human senses to sort species into kinds. So we read from Genesis chapter 2 that Adam was in fact able to name all animals. So and since then all animals corresponded to different kinds. We had not yet had any speciation. He was able to identify kinds. And similarly, Noah was able to get animals of each kind into the ark. So that suggests that he was able to recognize them. And that also, since we are descendants of Noah, we probably can do this as well. And then, so let's apply the cognitum method to the cat family. So we see here from different members of that family, the cat, the jungle cat, the bobcat, the cougar, leopard, and tiger, they do have strong visual resemblance. And we already know from the hybridization experiments that they seem to belong to the same kind. And then the visual resemblance supports the conclusion of the hybridization experiment that Felidae is in fact a kind. But apart from hybridization and the cognitive method, there's a more modern method, statistical baraminology, and in order to identify kinds. And it's used for large, either morphological data sets having to do with the forms and shapes of organs, or molecular data sets. And these data sets are so large and of the kind that you cannot analyze them using human senses alone. So we need algorithms and statistical methods to do that. And let me explain basically how statistical baraminology works. So it has three major steps. So first you need to find a long list of characters. In this case I only have 10 characters, and they could be morphological or genetic. And then for each species you list whether that particular character is present or not. So the filled circle represents characters that are present for each species, and unfilled circles means that they are not present. And then in the second step, you compute a distance between each pair of species based on the fractions of characters that differ. So we see here that the distance between species 1 and 2 is 0.1. And that is because it's only 1 out of 10% of the characters that differs. Character 6 is present in species 1, but it's absent in species 2. Whereas if we compare species 1 and 3, the distance is 0.9, because they differ in all characters except character 6, where that particular character is present both in species 1 and 3. So 9 out of 10 character differs between species 1 and 3, 90%, and that gives us this distance. And based on this table of pairwise distances, we can cluster the species into groups, and that's called a heap map, because large. So in this case, this suggests that these 4 species are divided into 2 groups. 1 and 2 form their own group, and species 3 and 4 their own group. So let me give an example of a heat map with a real and much larger data set. And it consists of 33 animal groups or species, and we have, instead of 10, 20,000 characters, which are really proteins. And here we have actually modern humans, Denisovans, and Neanderthals. And this heat map suggests that humans form its own group. I will return to this example later, but it's it's very strong evidence for humans being unique, using a huge data set. So that was the first goal of baraminology, trying to identify. Yes. Yeah. It's a very large data set with 20,000 proteins. So different proteins are active in different tissues in the human body. But I will return to this. But these authors, they use sequence alignment methods to identify the proteins, and it could be all different kinds of proteins. But because different species can have the corresponding genes located on different chromosomes. So it's quite complicated to pair, to find the gene in one species that corresponds to another gene that codes for the same protein in another species. The answer is that it's a large variety of different proteins. Yeah. And then we go to the second goal of baraminology, to find the mechanisms of divergence and speciation within each kind. And as I mentioned earlier, a biblical motivation is that God created each kind to rapidly adjust to changed environmental condition. And in God's original creation, we can think of two major features. And here I have illustrated this with the created kinds. Here we have the first generation. Here we have 4 species today. So we have 3 speciation events where new species are formed. And then we need mechanisms of change in order for those speciations to happen. But we also have created diversity in the first generation. And so God created the first generation of organisms of that particular kind, like a Swiss Army knife, where including the possibilities to adjust to environmental changes in the future. And then based on that created diversity in the first generation here, this design mechanisms of change could take place and cause speciation later on in the history of this particular kind. And that implies that in many kinds we have lots of species today, and that means that the observed degree of speciation in each created kind is possible within a young Earth creationist time frame, because these created diversity and the design mechanisms change. They are very effective, so we can have speciation taking place very quickly. So contrary to what macroevolutionary biology believes, that it has to take a long time. So speciation within a kind, because it's designed, can happen very quickly. But then we also have another evolutionary non-design mechanism of change, and they are much less efficient, and these are deleterious changes. Most well known are mutations from one generation to the next, like copying errors, and we can think of them as an effect of the fall of man. So God's creation was perfect, but then we have decay in nature after the fall of man, and they can also actually, to some extent, cause speciation. We can have chromosomal rearrangements and things like that because of this genetic decay, and that can lead to speciation as well. So let's look more closely into, first of all, the created diversity. And this is really a topic of its own talk, so I will only go through this quickly. So I will talk about created diversity as the first example of created heterozygosity as the first example of created diversity. Then there is something called variation-inducing genetic elements or transposable elements, and introduced by Barbara McClintock. But creationists Todd Wood and Peter Borger, they saw that they were important also as a creative diversity factor. We also have epigenetic codes and structures that are important for epigenetic changes to take place later on. Tommy Alto is an expert on this. He gave a talk on this yesterday. But we also have redundancy. We know that some plants, for instance, they have two systems of photosynthesis that work for different climates, and they can be switched on and off. So it's like God's foresight in the first generation. And then we have engineering principles. Randy Guyutsa has written about this. We have, like in the cells, there are sensors, and based on these signal molecules from the sensors, we have logical circles, and then we have actuators that make these changes happen, often through epigenetic mechanisms. But all these engineering principles, they were part of the first generation. So these are different examples of God putting variation into the first generation in order to later on have quick speciation. So let us look slightly more into created heterozygosity. So we know that humans, for instance, have 22 pairs of non-sex chromosomes. So here we have Adam had 2 copies of the first pair. Adam had 2 copies of chromosome 1, and so on, until 2 copies of chromosome 2, the same for Eve. And we know that each DNA molecule is a double-stranded string of nucleotides. So we have a number of base pairs. So suppose at a particular position, Adam had, for one of his copies of chromosome 1, he had a C along the coding strand, whereas he had a T along the coding strand of that position for the other chromosome. Then that is called a single nucleotide polymorphisms. And God could have put lots of such single nucleotide polymorphism variation into Adam and Eve in the first generation. And this is not like mutation. So it is neutral variation that leads to diversification among the offspring later on. Then we have the second principle, variation inducing genetic elements or transposable elements. This is one example of such an element, a transposon. So this is part of DNA from one chromosome that is transferred to another part of the chromosome. And that was, the possibility of this was a part of the first generation in order to later on in the history of the kind make it possible for speciation to take place. And we have epigenetic structures, and these are histone proteins, and the DNA, the DNA molecules are wound around a collection of those histone molecules, and then we have codes like molecules attaching to those histones, and that determine whether the DNA string will open up at that place, and then those genes there will be expressed to proteins. And the codes for that to happen, they needed to be present in the first generation. And that, as I mentioned, then we also have redundancy and engineering principles for environmental tracking as 2 other examples of created diversity in the first generation. So that was created diversity. Let's now look at the mechanisms of change that lead to speciation. And they can be either designed or non-designed. We have both these types. So we have, for instance, we can have a genetic drift and founder effect. So when a new population is founded at an island, then the descendants, their genetic makeup will be influenced by the particular makeup of the founders. And that can sometimes lead to speciation. And so that's a purely genetic effect. So it could be designed, but we could also have non-designed. These founders could have some inheritable diseases that are spread to the offspring. And mutations, we usually think of them as non-design. They are copying errors. But there could be some design mutations. For instance, in the human in the immune system, we need lots of genetic diversity. And some of those mutations can be designed, actually. Then we have crossovers, and that is for two-sex organism, we get half of our DNA from the mother and half of from the father, and crossovers are points on switches between grandmaternal and grandpaternal inheritance. And that's part of God's design, because he wants more variation in the offspring. Then we have natural selection, and so it's we think often of nature doing the selection, but also individuals can sometimes decide to move to a new place and start a new group, like having a founder effect. And then maybe the most important of all mechanisms of change, the epigenetic changes, because they are like switches above the DNA level that can turn on and turn off genes, and epigenetic changes can happen very quickly. There are many types of epigenetic changes, and they are usually reversible as well, whereas some of these genetic changes are not. Then we have also developmental plasticity, changes during embryological development. Usually, it's very harmful to have mutations during embryological development, and the fetus will not survive. But the researchers have found that there is still some plasticity in embryological variation, and some creationists have suggested that that could also lead to some speciation. So that was the second goal of baraminology, to find the mechanism of change within each group. Let's now turn to the third group to find the ancestral history of each kind. So suppose that a kind has been identified, and that's the first goal of baraminology, and then we want to find the ancestral history. And that includes biogeography, often due to environmental changes. So creationists often believe that plate tectonics took place during the flood. And of course, that caused continental drift, and that also influenced the history of that particular kind, geographic spread. We also have animals rafting on debris, like pieces of wood and things like that. They could also explain geographic spread, but also climate variations, like the Ice Age a couple of hundred years after the flood, also influence the geographic history of each kind. But we can also look at this genetically to find the history of each kind, and then we can use mechanisms of change, in particular in DNA, to trace the history of each kind. So for two-sex species, and like for humans, we have only men having Y chromosomes, and so this is a Y chromosome tree for all men alive today. So the men alive today correspond to the tips of the tree, and then somewhere here in the middle we have the most recent common ancestor of all men. And that gives us, we could do similarly for other kinds and build their history for kinds with men and women. We could build Y chromosome trees. And it's also the case that mitochondria, these are the power or energy stations of the cells. They have their own DNA. It's only inherited from the mother. So we can similarly build, based on mitochondrial DNA, we can build female trees. So the tips are all women alive today, and somewhere in the middle we find the most recent common ancestor. And amazingly, it turns out that these trees, the time scale of these trees, both for the male and female trees, are consistent with the young Earth creationist time scale, less than 10,000 years ago. How long does it take to reach the most recent common ancestor? Due to work by Nathaniel Jensen, 2013, and Nathaniel Jensen and Jason Lyle from 2016. So let's look more closely into the human Y chromosomes, or mitochondrial DNA tree. So, as I said, for the human Y chromosome tree, the male tree, the tips are men or women for the mitochondrial DNA tree alive today. And then we have some key questions. So data only gives an unrooted tree. We don't know where along this tree the most recent common ancestor lived. So we would like to find the root of the tree. Another key question is, how fast do the branches coalesce, go together? Because that is related to when did the most recent common ancestor live. So let's look more closely into the age of the most recent common ancestor of the tree. And that is related to mutations. And let's look here at the branches of a Y chromosome tree, a male tree. They differ because of mutations. When a father passes on Y chromosome to his son, or the mother mitochondrial DNA to her daughter. And branches that differ by fewer mutations merge more quickly. And how quickly depends on the mutation rate and the generation time. So here we have an example. So here are 5 men alive today. 23 mutations are unique for this man, 28 for this man. 18 are located in their 2 chromosomes, but not in the others. And continuing in this way, you can build this tree. That's the principle for how to do it. Let's assume now that the root of the tree is located here. So we have 25 mutations to the left of this arrow and 61 to the right. And that makes it possible to build a rooted tree, actually. Because then we start from here, and then we have 25 mutations here because before we have the first split here, and so on, and then we get an unrooted tree once we find the root of the unrooted tree. So that's the principle. But we need, when did this founder live? Then we need to know the mutation rate. And the mutation rate is the number of mutations per generation. And the faster it is, the more recently the most recent common ancestor lived. And now it's only the last couple of decades that the mutation rates has been estimated with accuracy using the so-called pedigree methods. And pedigree methods is basically comparing, in this case, the Y chromosome between father and son, and count the number of the differences. And having a large number of father-son pairs, you can estimate the fraction of your mutation rates per generation. And this is a key point. It's much higher than previously believed, when the mutation rate could not be measured in this way, by at least an order of magnitude higher. And that implies that the age of the most recent common ancestor is of the order 5,000 to 10,000 years for a wide variety of species. And it was believed to be more than 100,000 years earlier. So let's now study humans in more detail. And I will argue that humans are unique, its own created kind, in 3 ways. Firstly, we will use statistical baraminology to argue that humans are genetically different from great apes and other species. And going back to this study I mentioned earlier, secondly, I will use non-sex chromosome data to argue that humans' genetic variations is consistent with ancestry from a first couple, Adam and Eve. And thirdly, we will use the female tree from mitochondrial DNA and the male tree from Y chromosome data to build humans' female and male ancestral tree with a time frame that is consistent with young Earth creationism. So let's start with this statistical baraminology study that I mentioned about, the paper by Leitner and Serrati from 2019. And it includes 4 cat animals, mice and rat animals, 2 of them, 18 monkeys and apes, including some great apes. And then we have modern humans, 3 groups, modern humans, Neanderthals and Denisovans. Chicken-like birds, 4 species, goose, ducks and swan animals, 2 species, and an African frog. And as I mentioned, this is a huge data set with 20,000 proteins, and for each species group, we mark whether that each particular protein is absent or present. And here, and then I go back to what I mentioned earlier. These proteins can be located on different chromosomes for different species. So you need so-called sequence alignment methods, where you need to align the same genes or corresponding genes from different species that might be located on different chromosomes in order to find the corresponding genes and hence the corresponding proteins that these genes code for. And the result is amazing. Here we have this heat map, and it reveals that humans are clearly unique, because here we have humans form its own cluster according to this heat map. And the 4 great apes, including gorillas and chimpanzees, they are more closely related to other species in this study than to humans. So it's quite because it's a huge data set, it strongly indicates that humans are unique. So let's now look at the second line of evidence to support that human uniqueness. And as I mentioned, we humans have 22 non-sex chromosomes, and the total length of them is about 3 billion nucleotides. And I mentioned about biallelic SNPs. These are positions where there are variations among humans. So suppose that we have one particular position here, a base pair, where in 40% of the human population we have C along the coding strand, so a base pair CG, and in 60% we have a T along the coding strand. So that's the SNP, that particular position. And then the minor allele frequency of a biallelic SNP is the frequency of the less common allele. So if C occurs in 40% of all humans at this position and T in 60%, then the minor allele frequency is 40%. So generally it's between 0 and 50%. And there are about 10 million SNPs with a minor allele frequency of at least 1%, and that's 0.3% of the genome. And the interesting thing now is that we can build an allele frequency spectrum, and that's an histogram of all of the minor allele frequency of all 10 million SNPs. So here we have all the 10 million SNPs, and how many are there with this particular minor allele frequency, and this particular, and so on. And the shape of the minor allele, of the allele frequency spectrum gives information about the history of humanity. So usually an old population has a higher fraction of SNPs with a small minor allele frequency, whereas for a young population it tends to be more flat. And for a creationist model, you can think of many of the alleles with a large minor allele frequency. They correspond to this created heterozygosity I mentioned about, God putting variation into the chromosomes of Adam and Eve. Whereas these SNPs with a small minor allele frequency, they correspond to more recent mutations. Whereas in a tree of life model, you don't have the concept of created heterozygosity. You only have mutation. So then these SNPs with a high minor allele frequency correspond to old mutations, and these ones with a small minor allele frequency correspond to recent mutations. And I published a paper with Anne Gauger 6 years ago, and there is another paper by John Sanford et al. where we fitted allele frequency spectra using chromosome data from chromosome 22 to a creationist model and an evolutionary tree of life model. And we had at least as good as fit for a creation model as for the tree of life model. But I think we have even stronger case for the creationist models using mitochondrial DNA or Y chromosome DNA. These female trees and male trees I mentioned about earlier. And I will tell you a little about the history of building up those trees. So in the 1980s, the first estimates of the age of the most recent common ancestor of the female tree was dated to between 100 and 200,000 years. The mitochondrial Eve, you have probably heard that name. And they depended on evolutionary assumptions, because basically you compared DNA of mitochondrial DNA among humans and then with chimps, and then assuming that the split was 6 million years ago, they estimated that the time of the most recent common ancestor for humans was 150,000 years ago. So this was based on evolutionary assumptions, so kind of circular reasoning. But in the mid-1990s, the first estimates of human mutation rates by the pedigree methods, I illustrated this earlier for Y chromosomes. It's the same principle for mitochondrial DNA. It's circular. And then we basically compare a number of mother-daughter pairs and check what is the fraction, average number of mutations per generation. And the first estimates of mutation rates by pedigree method, they gave conflicting results, but generally the age of the most recent common ancestor was found to be at least 1 order of magnitude smaller, because the mutation rate was 1 order of magnitude larger, and hence the age of the most recent common ancestor at least an order of magnitude smaller. And then in 2013 and 16, Nathaniel Jensen, he pooled all these studies and obtained a most recent common ancestor less than 10,000 years ago, consistent with Eve. He also identified 3 major branches from the root, and that could correspond to the 3 daughters-in-law of Noah. Because if you take the female line, it doesn't pass the wife of Noah, because then it goes through the sons, and this is a female tree. So that is consistent with it. It's quite amazing. And then if we look at the Y chromosome human tree, it's a similar development. In the 1990s, we had the first estimates of the age of the most recent common ancestor, and that and those gave numbers several 100,000 years based on low-quality DNA data. Then in 2019, Nathaniel Jensen and Holland, they pooled several studies with high-quality DNA, and they estimated the mutation rate with, again, the pedigree method, which is much more reliable, really more empirical, comparing DNA of fathers and sons. That gave a most recent common ancestor living about 4,500 years ago. And according to the Bible, all men are descendants of Noah. That is the most recent common ancestor, not Adam. And that is amazing that you get this number. And then a couple of years later, Nathaniel Jensen published the book Traced with a young Earth creationist timescale of the Y tree. So also secular scientists, they build the same unrooted tree. So everyone agrees on the unrooted Y chromosome tree. As I mentioned, the question is where to put the root and how fast do the branches coalesce. And in this book, he managed not only to identify the root 4,500 years ago, but also 3 branches merging from this root that then correspond to the 3 sons of Noah. And also later on, the early branches of the tree, they could correspond to descendants of Noah a couple of generations later on that we read in Genesis chapter 10. That's then are sort of founding different subpopulations over the world. So let me end with some summary and conclusions. So the study of created kinds, it involves 3 major goals. Firstly, to find all created kinds. Secondly, to find mechanisms of divergence and speciation within each kind. And thirdly, to find the ancestral tree history of each kind. And this is really an intensive field of research. And as I have hinted at during this talk, it includes lots of new molecular data, genome-wide data, ancient DNA from species not alive today. We know that we can sequence DNA from Neanderthals, for instance, and we also have many sophisticated tools. I mentioned statistical baraminology, and they were based on sequence alignment methods to identify proteins from different species that could be located on different chromosomes. We also need tree building algorithms in order to build these trees and so on. And the results, they clearly show that the data group species into kinds. There still remains a lot of work to exactly identify all biblical kinds, but generally data really show a clear grouping of species into different kinds. And we have designed mechanisms of change that allow for quick speciation within kinds, which is consistent with the young Earth creationist timescale. Speciation can happen very quickly. And about the history of each kind, we can use mitochondrial DNA for female trees or Y chromosome DNA for male trees for species with men and women or two-sex species, and find that those kinds have a recent ancestry, also in agreement with the young Earth creationist timescales. And then we looked, at the end of the talk, I looked at humans, and the conclusion is that humans are unique using genetic analysis, and that also confirms the Bible and also human history. And my prediction for the future is that future research, based on even more data, we have large consortia like the Biogenome project, where the goal is to sequence basically all species, to sequence their DNA, and then we get even more data to sort of identify the biblical kinds. That this analysis of this new data will continue to strengthen the bushes of life model and of human uniqueness. And hopefully more people will realize how reliable the Bible is, because this is really described in the first chapters of the Bible about the biblical kinds, and that in turn will lead them to give their lives to Jesus. And let me end with a quote from the book of Romans, chapter 1, verses 19 and 20. Then the Apostle Paul writes, what may be known about God is plain to them, because God has made it plain to them. For since the creation of the world, God's invisible qualities, his internal power and divine nature, has been clearly seen, being understood from what he has made, so that people are without excuse. So we can see God behind what he has made, and that also includes that he created animals and plants after their kind. And we can see this from his creation, and even more clearly when we analyze data, recent data that scientists bring forward to us. And thanks for your attention.
Thank you, Ulla, for sharing your knowledge about baraminology. And yeah, we have now a break for just 30 minutes. And yeah, please have a look at the time that you're in time for the next session. Thank you. Okay, I think we can start. Good. Yeah, welcome to our second session here for today. And Mark Harwood will guide us today in understanding the importance of being open for new discoveries and following the evidence wherever it goes. And yeah, this session will go until 12:30, and then we will have a break for lunch. And yeah, and now I will hand over to Mark Harwood, and thank you for being here and sharing your knowledge with us.
Thank you. I'll just use it. Well, good morning, everyone. It's wonderful to see you all here again. You know, Christians are often accused of ignoring the scientific evidence. And as an engineer, I always like to go where the evidence leads because it makes sense to me. I like to understand the world that I live in to the extent that I can. But I think that the evidence is in fact very much in favour of what God's word says. And I believe that because I believe God's word is the truth. And in fact, it's the only source of reliable truth that we have. Everything else seems to be man's opinions. And so man's opinions are going to be driven by what people actually believe. Now, I think we see that evidenced in our society today. And I like this illustration of the two castles, and it kind of illustrates the battle that as Christians we find ourselves in this secular society of ours. So on the left-hand side, we have the humanist castle, the secular humanist religion of the world, which is based on the assumption, firstly, that there is no God, and therefore man alone decides truth. And man's story about how the universe came to be is, of course, the evolutionary story. On the right-hand side, we have the church, or Christians. And the foundation that we have is, of course, that there is a God, that his word is true. And in the opening chapters of Genesis, he tells us all we need to know about how he created this universe of ours. Now, coming out of the humanist castle are all kinds of social issues that we face today in our society. And the humanists are very busy at attacking the foundations of the Christian faith, which is the opening chapters of Genesis, where God tells us about the creation, perfect, without any blemish, could not have been better, and how man, made in God's image, rebelled against God and brought suffering and death into the world. The Christians, some of them are actually attacking the social issues, which is all well and good, and so we should. Unfortunately, some Christians are busy trying to destroy the foundations of the Christian faith by arguing that maybe we should accommodate the millions of years of evolution into our Christian understanding and our Christian worldview. Some Christians are actually lining up to take shots at other Christians, which is very sad. Some Christians are just firing off into space, really no idea what they're doing at all. And others are absolutely sound asleep and have got no idea there's even a war happening around them. So what is the solution to this problem? Well, I think the solution looks like this. First of all, we need to start and rebuild the foundations of the Christian faith. We need to return to the Bible as the Word of God, base what we believe on what it says plainly and clearly. We need to be attacking the foundations of the secular humanism castle, the society's belief system. We should, of course, continue to address the social issues that we have. But the first step is to rebuild the foundations. And you know, the Bible says, and I've shared this scripture before, it's probably our key scripture at Creation Ministries International, always be prepared, but I've used a different one, I've just realised. We demolish arguments and every pretension that sets itself up against the knowledge of God. And that's, I guess, is the same message as 1 Peter 3:15. So the arguments and pretensions that we come against today are things that stop people believing that God is real and that he exists and that we should be worshipping him. And I think one of the greatest pretensions that stop people coming to a knowledge of God is belief in evolution. Because after all, if evolution is true, how do we know there even is a God? Now, how do we demolish the arguments? Well, that's all about being aware that there are answers to the challenges that our society makes to the Christian faith. And I've already shared with you our wonderful website, but I'll do it again. There's a section in there on other languages. There are many articles that have been translated into German. We have a search window which gives you access to a vast array of wonderful articles, some very technical, some written at lay level, covering science, theology, philosophy, history, and so on. And there's a new article on the front page every single day. And I'm sure you all know what I'm going to do next. I'm going to ask you to say the name of the web address when it comes up onto the screen. So is everybody ready? So if you want to know anything about creation, you just go to... Very good. I think by the time I get to the last talk, everyone will be right onto that. So I had the great privilege of working in the aerospace industry as a design engineer. I then moved into the commercial side of the satellite business in Australia, and I was the general manager for strategy and planning for the satellite operator. So the kind of science I was engaged in is what you could call experimental science. That's the kind of science that depends on observable, repeatable experiments, the things that we can do, observe, and they tell us how the universe works. But as I've shared, some of you would already know, there is another kind of science. It's called historical science. And in historical science, the scientist looks at evidence in the present, but then he makes up a story about the past to explain what he's observing in the present. So this man is examining this rock and he's discovered a couple of bone fragments in it. And so he wonders to himself, how did these bone fragments fit together? So he uses his imagination. Perhaps they were part of a head like that. And then in this particular case, he thought, I know what this is. This is the missing link between land mammals and later sea mammals. You see, in the evolutionary story, life evolved first in the sea. Somehow or other, we came up onto the land and acquired legs and so on. Land-dwelling mammals finally evolved. And then, somehow or other, the land-dwelling mammals went back into the sea and became things like whales and so on, the sea-dwelling whales. Now, we didn't just make that up. That actually comes straight out of a paper, and that's the drawing that was submitted. And the authors were very, very confident. They said, in time and in its morphology, which means its shape, Pachycetus, this is the creature, the name they gave the creature, is perfectly intermediate, a missing link between later land mammals, earlier land mammals, and later full-fledged whales. Now that's a very confident statement, isn't it? It's perfectly intermediate. By the way, Pachycetus is Latin. It simply means a whale found in Pakistan. So this was considered to be the missing link between land mammals and whales. And you can see it's sort of got leg-like flipper things here, and it's diving in, chasing after some fish. Now, after some 7 years or so, by the way, just remember, what they found was just these samples, a few teeth and a piece of jawbone and a piece of the skull. And on the basis of that limited evidence, that whole story was told. Now, 7 years later, they found a full skeleton of Pachycetus, which looks like this. And so now they think he looks like that. So you can see that as more data comes to hand, there is less room for imagination. And so Pachycetus, as a missing link, vanished, although it still turns up in textbooks. So let's summarise that. Experimental science is about the present. It's about observable and repeatable experiments. That's how we know how the universe works, how we invent wonderful gadgets and devices like mobile phones, computers, and the like. Historical science is about the past. It's about unobserved, unrepeatable things. And it's in this area, what I like to call the storytelling science, where the conflict occurs between God's word and the claims of the scientific community. And no wonder the conflict occurs here, because in the evolutionary side of historical science, people begin by assuming there is no God, therefore we must find naturalistic explanations for everything. But of course the Bible says there is a God, and this is the area of the conflict. So science studies repeatable things, but it's history that studies unrepeatable things. Let me give you an example. We hear much about the ages of rocks and fossils and things, and often the numbers are given with much precision. But come with me on a thought experiment. Let's imagine you've just come around the corner of your house into your backyard, and you find a bucket partly filled with water underneath a dripping tap. Now, because you're scientifically inclined, you can't help yourself. You measure the volume of water in the bucket, you measure the rate at which the tap has been dripping, and then you ask yourself the all-important question, how long has the bucket been under the tap? Now, I know it's before lunch, but I think we should be able to do some arithmetic here today. So who can tell me, if you have 6 litres of water in the bucket and it's been dripping at half a litre per hour, how long has the bucket been under the tap? Anybody like to volunteer? 12 hours, yes. Who thinks 12 hours? Yeah, seems pretty reasonable, doesn't it? But remember, we have just chanced upon the scene, right? We were not there when the bucket went under the tap. So to get 12 hours, we made some assumptions. Who can tell me what we might have assumed? Other than you? No, yes, yeah. Yep, you have to assume it hadn't rained. I mean, if we did it today, you'd get the wrong result, wouldn't you? Yes, anything else? We started with an empty bucket. But how do we know that? You see, we weren't there and we didn't see the bucket put under the tap. So that's an assumption we can't prove. Another one, perhaps? Yeah? The tap's been dripping at the same rate. What if somebody had turned the tap on hard, partly filled the bucket, left it dripping and then walked away just seconds before you came around the corner? You see, your 12 hours would be completely wrong. But it turns out that your backyard is a very important backyard, and it has a resident historian. And this historian faithfully records all the events of your backyard. And the historian gives you a document, and the document says that at 1:05 p.m. the bucket was placed under the tap. At 1:50 p.m. you came around the corner. So now the question, how long has the bucket been under the tap? Who would like to have a go at that? Yes? 45 minutes. But I might say to you, but you're just a historian. I'm a scientist. And I have wonderful technology that can measure the volume of water in buckets by counting the very molecules. And I can measure the drip rate of taps down to, I don't know, millilitres per month or something. And when I apply all this amazing technology, well, actually, you're right, it's not 12 hours, it's 11.973621 to the nearest microsecond. So who thinks that with all the amazing technology, the scientist is closer to the truth? Oh, not a soul. Very good. Who thinks the historian is closer to the truth? Who thinks I'm trying to trap you? Well, the fact is the historian wins every time, because the historian is the eyewitness observer of what actually happens. And he gives us a document telling us, we presume, the truth. So let's summarise all that. Age can't be measured. You see, scientists look at the physical and chemical properties of their samples, and they can measure things, but they cannot measure age. Ages are always calculated, and you know you can get any result you like depending on the assumptions that you make. But the worst thing is, you can't verify the assumptions because they all occurred in the past, and we can't go back into the past to make measurements. So the age of something can really only be determined accurately by a reliable historical record. So the next time you hear a statement about a rock being so many millions of years old, think to yourself, what is the assumed history behind that number? What is the historical basis? Because it doesn't have a scientific basis, it has a historical basis. So how do we find out the truth about our origins? Well, it's a bit like the scene of a crime. We need to have an eyewitness, someone who is there, someone who knows everything, who loves us, who would not deceive us, and who has written down everything we need to know about our origins. And friends, we have that in this book, the Bible. Now, the Bible outlines a history of the universe. And we all know this, but let's just summarise. God created the universe in 6 days, around 6,000 years ago. Man's rebellion brought death into that very good world. God judged the world with a global catastrophic flood. God's Son Jesus came and died to pay the price of our sin. But he rose from the dead, proving he is God's Son. And Jesus will return again at the consummation of the age. So we have a whole history book which outlines the key essence of the gospel. Do you know the Christian gospel rests on and depends upon history, actual events in this space-time matter universe? So I want to ask you a question this morning. Is your faith supported by evidence? Now, it's not that our faith is based on evidence, but we need supporting evidence. And as an engineer, I look to make sense of the world around me. So I want to use two images today. The first one is of a little girl taking a tentative step into this pond or lake. She can see that there's ground underneath. She's not going to fall into a huge hole. But nonetheless, it still takes a little bit of faith to take that step into the water. But it's supported by evidence. And I want to use this image to depict a leap of faith. I just hope that this young man has checked that the water is deep enough and there are no rocks around. And this would be faith in spite of the evidence, or perhaps even contrary to the evidence. And I want to look at a number of different aspects of science and check out where the evidence actually leads. Now, if you were in outback Australia, stepping carefully around the snakes and spiders, it's not really that bad, and you came across an old rusty car like this, you would know that somewhere in the past there must have been a beginning. It rolled off the production line, bright and shiny, but here it is, deserted and ruined. There had to have been a beginning because it's running down, but it hasn't yet completely run down. You know, our universe is exactly the same. The universe, we discover, is winding down. Stars are burning up all their thermonuclear fuel. It's being dissipated as heat. And cosmologists tell us that the universe will experience a heat death. And there had, therefore, to be a beginning because it's running down, but it obviously hasn't yet fully run down. This article was published in New Scientist, and they said, this is the death of the eternal cosmos, because there were competing ideas that maybe the universe is of infinite age, it has always been, or perhaps it had a beginning. And the point they make here is that space-time can't possibly be eternal in the past. There must be some kind of boundary or beginning. Now, the beginning we are told about in our culture is the Big Bang. And the story goes something like this. There was an explosion about 13.8 billion years ago when absolutely nothing became absolutely everything, initially in the form of a hot plasma gas, some of which condensed down and formed stars and then galaxies, and some of the stars burnt up their fuel and exploded and became supernovas, and more complex molecules began to be formed, and finally planet Earth emerged, and then here in the lower right-hand corner, this amazing double helix molecule containing all the instructions for making every living thing arose all by chance. So this is the Big Bang story. But, you know, intuitively, it really doesn't stack up against observations. I mean, last time I saw an explosion, it just left a field of debris scattered everywhere. It did not produce highly structured, ordered results. And I like this cartoon a bit tongue-in-cheek. The lecturer is saying, now students, hydrogen is a gas which, if left long enough, turns into people. But let's just check in everyday life. Do we experience an explosion bringing order and structure? Now, this next image will come as a shock to many of you. And I want you to understand that this is not my daughter's bedroom. But you may never have seen anything like that. Is that true? Well, if you wanted to address that problem, you would ask the young lady to leave the room, and then you could create an explosion so that suddenly it would end up looking like that. But of course we know that's not what happens, is it? First of all, you require presumably some discipline, and then some hard work and intelligence to arrange everything so it's nice and neat. So here's an interesting example of a very cleverly designed machine that takes the raw chemical energy in the bonds of petrol and converts it into mechanical energy, which, driven through a gearbox, propels your motor car. But if you were to take exactly the same fuel and pour it over the top of your car, you get a very different result. So this is an uncontrolled application of raw energy in an unintelligent and undesigned environment. You see, order cannot arise just from energy alone. I've often come across the argument that says, well, the Earth is not really a closed system. The sun is out there and it pours down its heat and light onto the Earth, and that's the mechanism by which order arose out of chaos. But interestingly, the Big Bang story fails at many, many levels, and I just want to share a couple with you today. First of all, the universe should not actually exist. Einstein's famous equation says there should be equal amounts of matter and antimatter. And matter and antimatter will mutually annihilate each other. So the question is, why is there any matter at all? Immediately after the so-called Big Bang, it should all have just combined again and back to nothing. The Big Bangers believe there must have been a tiny imbalance of the order of one proton of another to produce the universe. So we need to have a bit more matter produced than antimatter. This is called the baryon asymmetry problem. So some very careful experiments were done recently at CERN, and they find that with enormous precision, parts per billion measurement, that they get exactly equal results. All of our observations find a complete symmetry between matter and antimatter, which is why the universe should not actually exist. So the observable evidence actually does not support this Big Bang model. You know, there's a real dilemma that the secularist has. If you ask the question, did the universe have a beginning, yes or no? Well, you've got two choices. If the answer was, well, yes, then something, everything, came from absolutely nothing. Excuse me, no matter, no energy, no space, no time, nothing. In other words, it had a beginning, and this is the prevailing view now with the Big Bang. But there's a problem here because this violates the first law of thermodynamics, which says that the total amount of matter and energy that exists can never change. Matter and energy can interchange, but the sum total is invariant. Okay, perhaps the answer is no. Well, if no, then the universe must be eternal. But now we violate the second law of thermodynamics, which states that in a naturally occurring process, disorder or entropy increases and energy is dissipated as heat. The universe, we observe, is running down. But since it has not yet run down, it must have had a beginning. So the problem is, whichever way we go, we end up violating one of the laws of thermodynamics. So the first law says that the universe cannot have had a beginning, therefore it must be eternal. The second law says that the universe cannot be eternal, therefore it must have had a beginning. So the secularist has a dilemma. Somehow or other, we must violate one of the fundamental laws of physics for the Big Bang to be possible. The only solution to that dilemma is that there is an external agent to the physical universe who was the creator, the causal agent sufficient for it in every way. So it's actually a huge leap of faith to believe that the universe made itself. But what a tiny step of faith to believe that the universe was made. Now, this history book of the universe gives us a timeline of events starting right from the creation week. And I outlined this yesterday, but importantly, from here all the way to Abraham is about 2,000 years. We know from history and also from the scriptures that from the time of Abraham to the time of Jesus is about 2,000 years. And of course, from the time of Jesus to the present day is about 2,000 years. So according to the Bible, here we stand today about 6,000 years after the creation. And 6,000 years just sounds preposterous in this culture, doesn't it? Because we're told over and over again about the billions and billions of years of Big Bang history. Well, the vast periods of time are a necessary consequence of the assumption there's no God. And so we're limited to natural explanations. And most of the so-called proof or evidence supporting that assumption comes from radioisotope dating. Now, my wife and I had the privilege of visiting the Royal Tyrrell Museum in Canada, and there was a sign inside the museum, and this is what it said: scientists use relative dating and absolute dating to determine the ages of rocks and fossils. Relative dating simply places events before or after one another, often without reference to time. Absolute dating uses radioactive properties of rocks and fossils to assess a more precise age. Well, does it really do that? I want to share with you an illustration. I've called it the dating game, and whenever I use this when there are young people present, everybody pays attention. But I have to point out, this might not work in German, I don't know if it does, but in English, dating means when boys and girls start to go out together. But this is a story around a lady called Kate Berensmeyer, and she discovered a volcanic layer called a tuff. And this created a great deal of interest because associated with this volcanic layer were fossils of elephants, pigs, apes, and tools. Now, the significance was that this was found on the eastern shore of Lake Turkana in North Kenya. So it was a very significant area where people believed human beings first began to evolve from hominids, or from the ancestor of humans, common with the apes, whatever they were. So it was samples were taken of this and sent off to Cambridge, to the world's then most advanced radioisotope dating laboratory. And back came the results using the potassium-argon method 212 to 230 million years ago. But there's a problem here because there were tools associated with this layer. And that's too old for tools, so they rejected the date. That can't be right. Now, notably, there was nothing wrong with the measurements that were done by the Cambridge laboratories and nothing wrong with the calculations, but it didn't fit the story. So they resampled the tuff, and they used potassium-argon dating of some feldspar and pumice, and they got an age of 2.61 million years. Now, that's almost a factor of 10 less. And they confirmed the data by paleomagnetism and fission tracks. So here we have 3 different dating methods that were lining up for the age of this tuff. A wonderful triumph of observational science. The problem was that Richard Leakey came along and discovered in 1972 a skull, a very famous one called KNMER, which I think stands for the Kenya National Monument. I'm not sure what the ER is, 1470. And this was a rather modern-looking skull, which was a problem because 2.61 million years is too old for this skull in the evolutionary story. So the date was rejected. But once again, there was nothing wrong with the measurements or the calculations. So they sampled some other bits and some more feldspar and pumice, and they produced an age of 1.82 million years. Now, that was a bit more acceptable to have a modern-looking skull. And here's the thing, they confirmed the results with paleomagnetism and fission tracks. Do you notice a certain irony here? And that date became the accepted date. But you see, it was the paradigm that determined which date would be accepted. Not the science, not the measurements, not the calculations. It was the belief system. And this is how human beings work. We have a worldview, we believe certain things, we interpret data in accordance with our worldview. And if we come across something which differs, we have a number of responses. Sometimes we just ignore it, pretend it doesn't exist. Other times we might park it and think, I'll have to consider that. There must be an explanation. Very rarely do we actually change our worldview. It's very difficult to change what we believe. That's why I say, particularly to young people, base what you believe on the only source of truth there is, the rock-solid basis of God's Word. Well, we looked at some other evidences in the world around us. The Niagara Falls are busily eroding away the gorge here, but it's been calculated at the current rates, which are around about 1.2 to 1.5 metres a year that the falls are receding up the river, that they would have all come to that stage in less than 4,000 years. The White Cliffs of Dover, they're eroding at a great rate, about a metre every 6 years. This is supposedly Cretaceous chalk, which is 65 million years, apparently that old rather. So the coast at that rate would have receded 8,000 kilometres. So the United Kingdom would have extended a long way towards the equator. Now, nobody's actually suggesting that it did, of course, but the point is that the observations are consistent with just thousands of years, not millions. When we look at our planet Earth, we have a magnetic field, and that magnetic field is presumed to be supported by a circulating current. But the problem is, measurements that have been made since the 1800s of the Earth's magnetic field show that it's decaying with a half-life of about 1,400 years. That means that at the time of Jesus, the magnetic field would have been more than 2.5 times stronger than it is today. And at the time of the flood, the magnetic field would have been about 9 times stronger than what it is today. But here's the problem. That stronger and stronger magnetic field would have required stronger current, which would dissipate more heat, which would place an upper limit on the age of the Earth of somewhere less than 20,000 years. Because back then it would have been so hot that life could never have begun in the first place. I've made reference to this article a number of times, and I do encourage you to have a look at it. The Age of the Earth, 101 evidences for a younger age of the Earth and the cosmos than what we are commonly told. Now, it's not that there are only 101 evidences, it's just that that sounds good. And so we have some in the wings, and occasionally we swap them out. But you can find it here at creation.com/age. So friends, it's actually a huge leap of faith to believe that the Earth is billions of years old, because the observable evidence does not support it. And that comes as a shock to many people, because all we hear about are those evidences which can be construed to support the vast age of the Earth. But it's just a tiny little step of faith to believe that the Earth is only thousands of years, because there is so much evidence in support of it. But what about this question of the origin of first life? This is perhaps one of the most profound issues that the naturalistic worldview has to deal with. Not only did everything come from nothing, which in itself is big enough, but inanimate chemicals somehow or other had to organise themselves spontaneously into the first life. And here is Professor Paul Davies. He's an Australian scientist and not a Christian. And he says nobody knows how a mixture of lifeless chemicals spontaneously organised themselves into the first living cell. And he goes on and says, how did stupid atoms spontaneously write their own software? You see, our cells are all controlled by a stored program on our DNA, which instructs the various cellular machines to do what they have to do, and so that the cell can function and so that you and I can live. In fact, if you were to take a cell and enlarge it to be the size of a great city like London or New York or Sydney, then you would find a bewildering array of technology inside. And it's extraordinary machines there, the temperature control systems, communication networks, transport systems, and so on. But not only that, it's capable of reproducing itself, of synthesizing or making proteins. Now, the protein synthesis process, and there will be a short examination on this at the end of the session, consists of let's say proteins consist of up to tens of thousands of amino acids, selected from the 20 different types in order specified by the DNA code. So the first step is called transcription, and a machine called an RNA polymerase reads the DNA. It produces a messenger RNA, which is then translated into, using ribosomes and transfer RNA, into that string of amino acids that we see at the bottom there, and when they're bolted together, they form a protein, and that then has to be appropriately folded. The first few letters we have there are actually of a protein called alpha HB, which has 141 amino acids in total. So biology students having to study this stuff before the age of videos must have been totally bewildered by all this stuff. I mean, that's meaningless. I don't know what that means. Maybe you do, but I've got no clue. But when you look at what these machines do and how they look, thanks to the wonders of electron microscopes, they are truly astonishing. Let's have a look at the process of protein synthesis.
One of the most amazing information transfer systems in the cell involves the way proteins are encoded in and decoded from strands of DNA. Starting out with these long, fragile, sticky strings of DNA, an amazing machine called an RNA polymerase zips down the strand, opens up the coils, and copies the DNA into another molecule called RNA. The RNA uses a slightly different language convention. It still has 4 letters like DNA, but instead of A, C, G, and T, it has A, C, G, and U. The RNA then leaves the nucleus and another amazing machine attaches to it. It's called a ribosome, and it is in charge of translating from the language of the RNA with its 4 letter bases to the language of proteins with their 20 different amino acids. 3 bases on the bottom of the tRNA match to 3 letter groups in the RNA. On the other end are amino acids which pop off as they're attached to the growing protein strand. But the process isn't finished because most proteins need help folding properly. And that's facilitated by little molecules called chaperones. They attach to and protect the unfolded protein as it's transported to a watermelon-shaped molecule called a chaperonin, which folds the protein into its final form. From DNA to protein is an incredibly complex process that uses precise and complex machines to translate between 2 completely unrelated languages, the linear code of the DNA world to the 3-dimensional code of the protein world.
How staggeringly complex is that, just to produce a protein? And Professor Sir Fred Hoyle, who is not a Christian but an astute scientist, said the probability of the formation of just one of the many proteins on which life depends is comparable to that of the solar system. Think about this, the solar system packed full of blind people, randomly shuffling Rubik's cubes and all arriving at the solution at the same time. I mean, can you imagine what that would look like? But it was back in the middle 1800s that Louis Pasteur formulated what we now call the law of biogenesis, that life only originates from life. And of course, that flies directly in the face of what the evolutionary story would have us believe. I want to share a quote from a very famous scientist, George Wald. He was a Nobel laureate. And he said, "One has only to contemplate the magnitude of this task to concede that the spontaneous generation of a living organism is impossible." And you think, hallelujah, he's got it. So here's a man, very intelligent. He looks at the problem and he says, it just can't be done. It's impossible. Wonderful. But then he goes on and says, yet here we are as a result, I believe, of spontaneous generation. So he just says it's impossible, but I believe it. Can you see the power of belief? Anything but confront the reality of a creator God. You know, when I look at that, I'm reminded of Romans 1:22, professing to be wise, they became fools. So, friends, I think it's actually a huge leap of faith to believe that life arose through time and chance alone. But what a tiny little step of faith to believe that life was created. Well, what about the evidence of the fossil record? You know, when scientists dig up the fossils, they find these things, but they don't have a little tag attached to them to say how old they are. Now, I know you all know that. It seems silly, but when you see the precision of the dates, you do start to wonder. But what they can do is what you would call the operational science. That's looking at the physics and chemistry of this. But when they make statements about the age, that's their interpretation. And that's historical science. It's based on the belief system of the scientist. But nonetheless, the fossil record does show a broad trend from marine creatures, so-called simple. Turns out they're not simple, but that's how they're viewed, all the way up to the higher level organisms. And you'll see down the side here, this scale all measured in millions of years before present. But the problem is that there is no evidence in the fossil record for this progression from simple to complex. And Darwin himself recognised this, and he said, why then is not every geological formation and every stratum full of such intermediate links? Geology assuredly does not reveal any such finely graduated organic chain. And this perhaps is the most obvious and serious objection that can be raised against the theory. So Darwin recognised this as a weakness, that the fossil record really didn't support his ideas. But he believed that as more and more fossils were found, that they would support his theory. So let's wind the clock forward 160 or more years. And a man called Colin Patterson wrote a book about fossils, and someone wrote to him about his book. He wrote back and he said this: I fully agree with your comments on the lack of direct illustration of evolutionary transitions in my book. If I knew of any, fossil or living, I would certainly have included them. Now this is a bit of an admission. This is a senior paleontologist at the British Museum of Natural History. But he went on and said, I will lay it on the line, there is not one such fossil for which one could make a watertight argument. But our children in high schools and students at university are told over and over again about how the fossil record supports this evolutionary development from simple to complex. But the experts acknowledge, well, actually, it doesn't. This was an extract from a paper published in Science Journal showing the evolutionary tree of dinosaurs. So across the top you have the different dinosaurs, the black lines here, the vertical lines. This is time, by the way, down the scale here on the left-hand side. They represent where the fossils are found in the presumed ages of the rock layers. Now, there are lots of dashed lines and so on. We're going to colour those in, but they represent the branches of the tree, but there is no fossil evidence for those. So if we take the red and blue lines out, what you suddenly see is that there is no tree. There's no fossil evidence for this branching tree of creatures. So the tree is, in fact, an article of faith. It's there because of the belief system in the evolutionary story. Now, I'm sure you've all seen this kind of an icon, the ascent of man, starting with the stooped ape and finally becoming more erect and ending up as Homo sapiens. I saw one recently which started like that, and then it continued on with Homo sapiens crunched over his computer. But anyway, apart from that cycle. But I want to share with you a video clip that was put together by a colleague of mine in Australia called Scott Devlin, and he examines the evidence for this icon of evolution.
You may recognize this image from school textbooks, t-shirts, soda adverts, and everything in between. But what if I were to tell you that if we're to be honest with the evidence that we have, this picture should look more like this? Let me explain. On the left is what is believed to be the first ape, known as proconsul, and on the right is a human called Homo sapiens. But what scientific evidence is there for everything else in this image? The transitional species. Let's find out. We'll start with Homo neanderthalensis. You might know him as Neanderthal man. Recent discoveries have shown that Neanderthal man made and wore jewelry, played instruments, used tools, and wore makeup. We've even found that his brain was the same or slightly larger than the average human living today. In other words, Neanderthal man was actually just a man. What about Homo erectus? Recent discoveries have shown that Homo erectus made tools, engaged in artwork, spoke intelligent language, and made and sailed boats. In other words, Homo erectus was also just a man. Now we'll come back to Homo habilis in a moment, but first, let me show you something. Have you ever heard of Lucy, the most famous so-called ape man, paraded in our museums, one of the very first Australopithecus afarensis species ever to be found? But what does the evidence reveal? She had a skull that was sloped and ape-like, nothing like human skulls, fingers that were curved, not at all like human fingers, toes that were curved, not at all like human toes, wrists that had the ability to lock for knuckle walking, and a knee structure that was compatible with life in trees. So Lucy and her kind swung from trees and looked like today's apes. Lucy is an extinct type of ape. As we've just seen, Homo sapiens, Neanderthal man, and Homo erectus were all men. Australopithecus afarensis and proconsul were both apes. Considering the ubiquity of the evolutionary icon, we'd really want to see some strong evidence for Homo habilis, the pivotal point of transition between stoop ape and upright man, between basic instinct and intelligent thought, between animal noise and intelligent speech. The only problem is, in the words of Ian Tattersall, Homo habilis is a wastebasket taxon, little more than a convenient recipient for a motley assortment of hominin fossils. Other scientists referred to him as a garbage bag because the bones we have for him are a mixture of human and ape bones. In other words, Homo habilis never existed. I think evolutionary professor Bernard Woods sums this up well. Our progress from ape to human looks so smooth, so tidy, it's such a beguiling image that even the experts are loath to let it go. But it is an illusion. With the lack of evidence and agreement on the ape-human transition forms, why is this not happening? Or this? And is a better explanation that man and apes have always coexisted and reproduced according to their own kind, as stated in Genesis 1.
I think he does a wonderful job of demolishing that famous icon, doesn't he? You can find that video clip, by the way, on Creation Ministries International's YouTube channel. Just search for "Where is all the evidence of the ape to man transition?" So I was rather taken by this quote. Now, evolution is the substance of fossils hoped for, the evidence of links not seen. Those of you who are familiar with the English Bible, that's a bit of an apology to Hebrews 11, verse 1. But we're told so much that we are genetically similar to chimpanzees, and there's less than 2% difference. But, you know, even if that was true, is that necessarily evidence of a common ancestor? You know, it turns out that we have about 50% the same DNA as a banana, but that doesn't make us half a banana. Some people think I'm bananas, but that's another point. Similarities are in fact evidence of common design, not common ancestry. You know, I came across this video clip from a secular website, and it's aimed at young people, and it analyses this question of how similar our DNA really is, and it's extraordinarily honest. So let's have a listen to this, remembering it's from a secular website. No, we're not going to be graced by that, unfortunately, and I don't understand why. Okay, so what they end up saying through that website, with that particular thing, is that if you ignore 18% of the chimp DNA and ignore 25% of the human DNA, then the remainder, yep, that's 98% the same. But that's extremely disingenuous because you don't hear that qualification made. People just say, oh, we're almost the same as chimps, therefore we have a common ancestor. But it's definitely not the case. So when we look at this so-called fossil record, all the way from simple to complex over vast millions of years, I think it is best explained by the order in which things were buried during the flood. So I think it's a huge leap of faith to believe that the fossil record records evolution over eons of time. But what a tiny little step of faith to believe that the fossil record shows the order of burial by the flood. You know, the importance of this issue is often recognised more by the atheists than by the theologians, which I find quite extraordinary. I want to share some words from Frank Zindler, who was in a debate some years ago, and he said this: "The most devastating thing, though, that biology did to Christianity was the discovery of biological evolution. Now we know that Adam and Eve never were real people. The central myth of Christianity is destroyed." He went on and said, "If there never was an Adam and Eve, there never was an original sin. If there never was an original sin, there is no need for salvation. And if there's no need of salvation, there's no need for a saviour. And I submit that puts Jesus, historical or otherwise, into the ranks of the unemployed." And he ended by saying, "I think that evolution is absolutely the death knell of Christianity." And friends, I think he's right. If evolution is true, we don't have a Christian faith. If evolution is true, the Bible is not. There was no original creation that was very good. There was no fall of man that brought suffering and death into the world. If evolution is true, there was no need for a saviour to go to the cross for us. You see, this issue strikes at the very root of the Christian faith, and we must defend it, not only with sound arguments theologically and philosophically, but also sound scientific arguments. I began with this quotation: "We demolish arguments and every pretension that sets itself up against the knowledge of God." How do you get equipped to do so? Lots of different websites around the world today. I'd recommend, of course, creation.com. We also have a presence on each of the 5 major social media platforms with a Creation Ministries international channel. And there's wonderful resources there that you can find. So friends, let me just end by saying that this issue is so central to the gospel that if you try and integrate the evolutionary story into your Christian faith, you end up with just a story which really has no meaning, and there's no coherent explanation for the sacrifice Jesus made for us on the cross. So my encouragement to you today is to, in fact, go where the evidence leads, because it all leads to the truth of God's word and the historical record that is laid out in it. And this book, of course, leads us to a knowledge of God and of his Son, the Lord Jesus Christ, and it shows us the way to salvation. So thank you very much for your attention. We have a few minutes left, so I think we can probably take questions if there are any. There's one down here, the microphone is coming.
Thanks a lot. We've talked about cells, and you yourself work in aerospace and the universe. My question is, do you find any similarity between these two systems that manifest God's creation?
Do I find any similarities in...
between these two extreme contrast systems, the small cells and also the big universe?
Oh, do I find? Yes, I do. I see evidence of design everywhere, from the smallest scale to the largest scale. There's clear evidence of design, as I've shared this morning, in the cellular structure and machinery and operation of every living organism. I see evidence of design in the construction of our Earth, first of all. I'm going to talk about the moon this afternoon and the role that it plays, but in our solar system, the position that our solar system has in the Milky Way galaxy is absolutely ideal to firstly provide a safe environment, but also to invite us to explore and discover how God has created the universe. So everywhere I look, because I use biblical eyes, starting from God's word, I see evidence that there is a God, that he's the creator, and of his character, that he's a God of love and compassion and mercy. And yes, I see that everywhere. Thank you.
Yeah, thanks a lot. I'm just curious, how's the situation in Australia? I was told yesterday by a brother from Ward and Wissen that since Corona, they as an organization have no access anymore to the universities. Do you have the opportunity in Australia to go to the universities and speak with people, or is it not possible anymore?
I don't think it. I'm not aware of coronavirus having changed the access. These universities are very secular, and you normally don't get onto a university campus other than through the invitation of a Christian student body. So, for instance, earlier this year I spoke at Sydney University, but at the invitation of a group of Korean Christian students. One of my colleagues, in fact, spoke at Curtin University in Perth several years ago now, before coronavirus, and the student group had advertised the meeting all around campus, and it certainly created quite a stir because there were a lot of people who came along with the expressed intent of disrupting the meeting. And there was amazing stories that emerged out of that. One that is a bit, I'm not sure I can remember it clearly enough to share, but it actually resulted in one of those antagonistic students coming to the Lord subsequently. But that's the only way you get onto campuses. I've had the privilege of speaking at a number of universities in South Africa. One in particular was kind of interesting. People found out that of my background in aerospace, and so I was invited by Stellenbosch University to come and speak to their electrical engineering department about spacecraft design, and from a commercial point of view as well as technical. They, of course, do all the technical stuff, and they wanted to know how do you blend this into a commercial model and make things work. So I gave this lecture. It was a lunchtime lecture, and then a hand went up, and somebody said, "Why are you here in South Africa?" Well, I was there on a mission tour, a ministry tour. So I explained what I did with Creation Ministries International. And the questions from then on were all about the character of God. Who is God? They weren't really interested in, business cases for satellites or anything like that. It's irrelevant. And one of the students, my wife told me afterwards, was pacing back and forth, shaking his head, saying, "I can't understand it. How could someone with intelligence believe in God?" But it's interesting, when I go internationally, often I can get into places like that because I'm a visiting international speaker. But in Australia, I'm just Mark Harwood, right? So it's something about the prophet in his hometown is without honour. But in secular education institutions, the doors are generally closed, other than, as I said, through student Christian groups. The same is true in state high schools. Interestingly, we can't get into some Christian schools in Australia because they receive funding from the government, and it's conditional upon adhering to a state curriculum, which expressly forbids the teaching of biblical creation, which is very sad to see that the Christian schools are being choked like that. But not all. There are quite a few of them where there is an open door, and if the door's open, we walk in. Thank you.
Okay, the time is running over. I think we have we take the time for one more question.
Thank you. Thank you for your talk. You spoke about that the Earth is losing its magnetic field, its energy. What will happen if the energy is totally gone?
The magnetic field, if that happens, then the magnetic field will collapse. But the magnetic field is a wonderful design provision because it deflects cosmic rays from the sun, and cosmic rays cause a lot of damage. They can do genetic damage. They have a whole series of other effects, but which, by the way, affects things like carbon-14 dating, because the magnetic field's decline means that there is more carbon-14 being generated through cosmic ray interactions with the upper atmosphere, which gives the impression that things dated using carbon-14 are older than what they really are, which means that the carbon-14 to carbon-12 ratio is not a constant. That's the point. So the Earth would become a very inhospitable place, but I don't believe that will ever happen, because the Bible tells us that Jesus is going to return for his bride, that company of believers, born-again Christians, and we won't have become some kind of mutated, distorted little creatures struggling to live. I think that'll happen. God will return. Jesus will return long before that. Indeed, I don't think it could be very far away, to be perfectly honest, just looking at what's happening in the world today.
Thank you.
All right, thank you very much for your attention again. Thank you.
Thank you, Mark Howard, for sharing your knowledge with us. Bevor wir zum Mittagessen gehen, haben wir noch ein paar Vermeldungen. Genau, und zwar haben wir jetzt die Pause bis um 14 Uhr und haben jetzt Zeit für das Mittagessen. Und genau, eine Erinnerung, die Experimente finden eine Etage über uns statt. Wer in der Pause noch Zeit haben will und Interesse daran hat, schaut es euch gerne an. Ansonsten haben wir noch einige falsch geparkte Autos, die am Busbahnhof stehen. Ich lese aber die Nummernschilder vor und wer sein Nummernschilder kennt, kann dann gleich sofort vor dem Essen einmal sein Auto umparken. Das ist einmal ELBE 392, NRPD 707, NRVF 134, NRP 1395, BOPV 7001, NRSV 237, NRME 417, NRAG 200. Ah, okay, ja, das war's. Dann, bevor wir zum Essen gehen, würde ich direkt hier fürs Essen danken und dann können wir sofort rübergehen. Ich will dir Danke sagen auch für diesen Vortrag von Mark Harwood, den wir jetzt eben auch hören durften, und ich will dich bitten, dass du deinen Namen ja auch hierdurch groß machst und bitte segne diesen weiteren Tag und danke dir für das Essen, was wir jetzt haben dürfen. Amen.
Gut, wir wollen pünktlich starten. Ein Hinweis noch, wenn es jemand von euch betrifft, wir haben 2 Autos, die noch vorne im Busbahnhof stehen. NRME 417 und NRSV 237. Also wenn jemand sein Auto da erkennt und vorne im Busbahnhof steht, ihr dürft noch umparken. Und dann darf ich nach dem Gebet auch direkt Toni Alto das Wort übergeben. Und ja, ich bete gerade. Pray.
Okay, many thanks for this wonderful opportunity to share this challenging, maybe, and of course fascinating insights on epigenetics with you. My name is Tomi Aalto, I come from Finland. I have a very diverse and interesting job history and background. I've been working, for example, in a German company called Festo in Stuttgart, Esslingen. In Finland, we have a Festo Direct and other sectors. I worked for Festo for 2.5 years. And so I've been working in automation companies, programming, making programming, robotic specialists, etc. But I'm also a teacher. Currently, I'm classroom teacher. So I have a master's degree in educational sciences in University of Turku, Finland. Year might be 1989 when I had my degree. So let's start this presentation by focusing on a couple of words and terms we can find and widely used in Genesis. And these words and terms, the first of them is the verb bara. It's a Hebrew word, and basic meaning is to create. But how is it used? This verb, it occurs 48 times in the Old Testament, and in the whole Bible, this verb is used about 243 times in different forms. So it's quite a common verb. The subject is always God. It's never used of human activity, such as making something, building, or crafting. And we have to be very careful when we investigate this verb. It's not an allegorical verb. It has a very specific meaning and purpose. The word doesn't always strictly mean creation out of nothing. So you may have heard the term ex nihilo. But it always refers to a new, unique, original, and supernatural act of God. And interesting is that the context determines whether it refers to ex nihilo or to creating something new in order of function. And here are a couple of examples how it is used. Genesis 1:1, in the beginning God created, then the verb bara is used, the heavens and the earth. And it's also used outside of the Genesis creation account in several other books, like Psalms, create in me pure heart, O God. The key idea is that bara emphasizes that God is the sole, original and unique and the only source of all creation. So it's a very person way and person type how this verb is used. So it's dependent in a person, Elohim person. Another word is a noun, the noun min, which has a basic meaning, a kind, category or type or a group or something like that. And it is also widely used in the creation account in several verses. And it has repeated in expressions like according to its kind. So this noun helps us to understand what a kind means, what a min means. It has semantic content. It indicates that God created plants and animals with diversity, but also with some kind of boundaries or barriers, so that each kind reproduces according to its own kind. The term is not identical with the modern biological term species. It doesn't mean that. It describes a broader reproductive grouping that maintains order within God's creation plan. And it also has its own context. The term emphasizes that created organisms don't cross into other categories. So a dog doesn't become a cat, or a duck doesn't become a sparrow, for example, or vice versa. But they remain with their God-given boundaries according to their kind or kinds. And so the key idea is that the noun min expresses both the diversity, but also the limits and the order that God has established in creation. And now I ask you to remember these, especially these limits and boundaries and barriers, because I'm coming back to these terms during my presentation. Yesterday I gave a presentation about epigenetics, and in this presentation I have a lot of same slides, so I think there's a lot of repetition, but I think it's not a bad idea to repeat some important things and understand how things work at genetic and epigenetic and molecular biological level. So let's start with a very known example of so-called evolution. This is, I think everyone of us knows this example. So Darwin's finches, this is his idea that there is natural selection and some kind of random changes. So according to Darwin, evolution and natural selection work as follows. Some finches develop strong and thick beaks as a result of random mutations, leading them to become seed eaters. Finches with slender beaks become insect eaters, and thus natural selection directs birds with different beak types to acquire more food, giving them a competitive advantage. They produce more offspring than birds that obtain less food, thereby reinforcing the trait in subsequent generations. This results in differentiation, which leads to speciation. And this is the foundation of the entire evolutionary theory. But today we know, and many even secular scientists know, that Darwin was seriously wrong, because this theory, it doesn't work anymore. It has never worked, but it was only a theory. But okay, Darwin makes some conclusions, but today we know that his conclusions were absolutely wrong. And next I'm going to explain this, why he was so wrong. This is a study, about 8 years old study, picked up from the Forbes article, and these statements are written by scientists. They write that these species of finch have distinct diets which could explain the differences in methylation patterns, as diet is known to influence epigenetics. They also know that those birds are genetically pretty similar, almost similar. DNA is almost the same. And they also know that DNA methylation can be a source of heritable phenotypic variation. And they also know that environmental stressors, such as pollutants, diet, etc., different environmental factors, can change these methylation patterns. And for them, this is it's a new mechanism, and yes, epigenetic mechanisms have been known for about 20 years. In theory, the idea has been known about 50 years, but through a true scientific level, the most important mechanisms have been known only for 5 years. So, but let's continue. They also know that the field of epigenetics is just getting started. So this is something new, and that is what we are going to study right now. Today we know that the size and shape of finches' beaks vary according to the type of food. So if a finch starts to eat seeds, then its offspring will have stronger beaks, and if it begins to eat insects, then after just a few generations, the offspring will have thinner and more delicate beaks. we know that these rapid changes are not based on random mutations or selection. That's absolutely an outdated theory. So evolution believers, they have to find another theory by which they try to defend their ideological foundation. Another excellent example of epigenetic regulation is the blind cavefish. Its widely has been used as evidence for evolution for decades, but is this based on random mutations and selection? No. But we still can find several articles on the internet which claim that over millions and millions of years these kind of changes happened and fish lost their eyes and probably the eyes don't come back and so on. But what happens in reality? The eyes are restored after the fish return to a light environment. And it doesn't take millions of years, only a couple of generations. So it happens very quickly. So there is some kind of epigenetic switch that turns to another position when fish live in a dark environment. So this sounds very designed, doesn't it? Because what does a fish, what does it do with eyes in a dark environment? It doesn't make sense to hold eyes in dark environment. So it's best to leave them without growing. So this exactly points to designed features. But the scientists were shocked when they realized that the eyes are restored after fish returned to a light environment, and especially that it doesn't require millions of years, it doesn't require thousands of years. This happens very quickly, after 2 or 3 generations only. Italian wall lizard is also a very famous example and so-called evidence for the evolution believers, but today we know that this rapid change also was based on changed diet, nothing to do with random mutations or selection. Here's the summary of the study. Scientists were almost shocked to discover how quickly the lizards have undergone radical changes. Their behavior had changed, their jaws had lengthened, and most surprisingly, their intestines had developed a ring-shaped muscle called the cecal valve. All of this had happened in just a few decades. The reason was clear. The lizards had to switch their diet from insects to plants. The scientists didn't find any consistent changes in the lizard's DNA. But as we can see, this was the new structure in their intestines. And how long did it take to grow this structure? What do you think? 30 years? 1 year? 4 million years. Yes, they say that. But in reality, it took only 1 generation. I've read a study that said that a lizard can grow the measure of its intestine in just 40 days. Just 40 days, 17% more of its length in 40 days. So these kind of changes, they occur very, very rapidly. And of course, evolutionary believers are shocked. They don't want to talk about these changes. I have few slides that I can almost skip very quickly because this presentation is quite massive. I have 65 slides, so let's go this through very quickly. But a bee larva grows into a queen because it's been fed royal jelly. The DNA is identical. Note. The ant grows into a soldier ant. Again, DNA is identical. No changes in DNA. Stick insect, they can grow wings, but nothing to do with changes in DNA, nothing to do with so-called mutations. Mammoths, different phenotypes, but again, genetic similarity is astonishing, based on epigenetic switching. Nautiloids, very different outcomes, but again, DNA is identical. These changes are based on epigenetic regulation. Butterfly metamorphosis, 4 different stages, but again, DNA is the same in all these 4 different stages. So it's about epigenetic control of gene expression. Human skin color, an example from 2016 from Great Britain. Amelie and Jasmine are monozygotic twins, means that their DNA is absolutely identical, it's the same, but they have a different skin color. So we can conclude that skin color is not determined by the DNA. It is controlled by epigenetic mechanisms such as alternative splicing. I'll come to that later. Different plants, these are variations of the wild mustard plant. So different sprouts, cabbage, kale, Brussels sprouts, broccoli. Virtually it's the same plant, different variations. DNA doesn't change. Or if it changes, then there are no explanatory changes that could explain these changes within these different species or different variations. Different birds. Wie können 9 Arten so unterschiedlich aussehen und dennoch genetisch nahezu identisch sein? Was that correct? Yeah. Okay, thank you. Maybe next time, yes, thank you. So, because I'm a teacher, I would like to provide with the most important question for a middle school or a high school biology teacher. And it's based on this fact. Every one of us in our bodies has about 300 different cell types. For example, skin cells, liver cells, etc. So there are only a couple of exceptions. All of our cells contain exactly the same DNA sequences, meaning that their genome is identical, of course, because we have started our life from one single cell that has started to divide to 2 and 4 and 8 and 16 and 32 and 64 and so on. So when the cell is copied and replicated, the DNA is, of course, also copied. That's why in every cell in our body is the same DNA. There are only a couple of exceptions. There are neurons in our brain. It's remarkable that in every cell in our brain there is a unique DNA sequence. It's incredible. Also, T cells used by our immune system, they might have different DNA sequences, but all the other cell types, they have exactly the same DNA. So now we can make the question, why does a skin cell have a different identity and function than a muscle cell, even though their DNA is exactly the same? So this is a basic question. It's a very logical question. But is this question asked for pupils and students in schools today? No. Why? Because evolutionary believers and supporters, they don't want to teach our students how the cell differentiation happens. Because if this was taught in our students and for our students, I think that most of our students wouldn't believe in evolutionary theory anymore. But this is not taught to students. So we can conclude that DNA does not determine the cell's function. And we can continue asking then what does determine the cell's function? And now we are coming to epigenetic regulation. But before that, I ask you to read this very important statement. I enjoy this glass of water. This is only 6 years old, but it's very important. Scientists have concluded that the nuclear genome has lost its function as the unique and exclusive determinant of inherited characteristics due to mounting waves of data revealing the complex epigenetic networks that heritably control genome expression. So they know that DNA doesn't control anything. There is another information layer called epigenetic information layer that controls how DNA is read and so on. And because we have 300 different cell types in our bodies, we know that there has to be some kind of differentiation or some kind of mechanism that causes these differentiation happen in our cells. So we have to ask why cells are different even though their DNA is same. We can have muscle cells, blood cells, all kind of different cells, but the DNA is the same. How is this done at the cellular level? Let's have a closer look. The word epigenetic, it consists of two words. Epi is a Greek word meaning on top of or outside of something. That means outside information that is outside of DNA. And that mostly means some kind of chemical layers of markers. For example, CH3 molecule, which is a hydrocarbon group, a methyl group, and this is called methylation. And they have a very important meaning and purpose. They direct the reading of DNA, so they control how DNA is transcribed. It means how it is read. They also regulate several number of cellular other mechanisms. They can silence DNA and prevent it from being transcribed. So by methylation, the cell can, in a way, suppress or hide, keeping them outside of reading. And again, by using different methylation or without methylation, the structure can be open and readable and allowable for transcription. And it's good to know that about 80, in some cases more than 80% of plants' genomes are silenced. That means that plants have a great adaptation potential. And we know that without these epigenetic profiles, markers, the cell is an undifferentiated stem cell. It has lost its identity, its function, its functionality, and its job. It doesn't know what to do. So we know that the cell needs epigenetic programming in order to perform its designated function. And also important to know that methylation stabilizes the genome. this is important information because viruses, they don't have their own methylation production. So they kind of steal the methylation from their host cells. And that means that the host cell loses its own methylation profiles. And it might result and lead to very serious diseases. Viruses are dangerous. Yes, they need methylation in their own gene expression, yes. But they don't have, they don't have their own methylation production mechanism. Yes, yeah. Mm. And they steal the methylation from you and me, from ourselves. What do you mean?
It's not always the virus would copy the whole string. It's not right. It's not correct that he had to copy the whole string.
Whole string of DNA. Yeah. Their own. Yes, they do. Yes. Yes, they have to do that because they have to copy themselves. Yes. But they copy, but they also read some sections of their own DNA. They don't read the whole section. Depends on virus type. Yes, yes. There are three main types of epigenetic information. The most common is the DNA methylation, and then we can have a look soon at histone epigenetic markers. They are very complex. And the third group is RNA signaling molecules. And mostly we are talking about non-coding RNAs. Here's the picture of DNA methylation. And we know that in eukaryotes, the methyl group is attached to a cytosine base, mainly at so-called CPG sites, which means that at the DNA sequence, the C base is followed by the G base. That is called a CPG site. The small p stands for phosphate. In prokaryotes, the methyl group is mostly attached to an adenine base, so they have a different system. Maybe, I think this is something to do, of course, with design. You might have heard in school times that might be some kind of gene in a genome, and one sequence of DNA codes for one RNA molecule that further codes for one protein. Are you familiar with this kind of idea? Now I have to say that we have... Yes, we have to forget this outdated theory. It doesn't work this way. Because the cell can produce thousands of different proteins from the same DNA sequence without changing the DNA. How is it done? Let's have a closer look. For example, we humans, our cells they only have about 20,000 DNA sequences used and read for protein production. It means it's less than an earthworm, for example, nematode, which has 22,000 protein coding genes. We have only 20,000 so-called protein coding genes, but the number of different proteins in our bodies might exceed 6 million. So you can make a short calculation that how many proteins on average are produced by reading only one DNA sequence. The answer is about 300. 300 different proteins are produced by reading only one DNA sequence. Incredible mechanism, isn't it? Das ist sehr gut. Yes. This is based on a very complex mechanism called alternative splicing, and it has epigenetic control and regulation by DNA methylation profiles, histone epigenetic markers, and non-coding RNA molecules. But this also makes us ask a question, what is a gene? Because actually there are not some kind of genetic units in the genome, informational units. There are no units. There are no genes in the genome. The gene actually is somewhere outside of the genome. I would say that the gene can be found at the RNA level. But at DNA level, we cannot find genes. There might be a very little share, a little minority, perhaps 1% something of our genome is directly encoded to RNA and further to a protein or other RNA molecule. So this picture might be quite difficult to understand, but I try to ravel it up. This illustrates how alternative splicing works. Please focus your attention on those colored segments. They are called exons, and the cell's mechanisms have selected them from different locations in the genome, even from different chromosomes. These sequences are joined together and transcribed into RNA molecule. The cell can modify this so-called pre-mRNA and in different ways by combining useful exons and removing unnecessary introns. In this way, the cell can create various combinations from the transcribed sequences and further produce different proteins. No modifications to the DNA are needed. This mechanism is epigenetically directed and regulated. And in this example, we have only 3 different proteins, but they are all of them are coded by reading only one DNA sequence. The maximum amount of different proteins based on only one DNA sequence has been found in Drosophila melanogaster, which means the fruit fly. Yes, and the number is 38,016 different proteins by reading only one DNA sequence. More than 38,000 different proteins. Sounds designed. So here it is, an example. It's an insect gene, DS-CAM, encodes more than 38,000. So we can now understand why organisms have so remarkable adaptation potential without changing DNA. So now we have learned that there are no single information units called genes in genomes. That's why most of scientists and researchers, they want to move the gene concept from the DNA level to the RNA level. It might be more sensible, and yes. We also can conclude that DNA actually is passive information data repository. It means that the cell picks useful DNA sections because it has a mechanism, but it is using microRNAs and other non-coding RNA molecules to find correct DNA sequences from the genome, and it picks and transcribes and joins them together to make a pre-mRNA molecule that is further possibly modified also. I'm not the only one who has concluded that DNA is passive information, sorry. So Dennis Noble, you may have heard of him, he said that the modern synthesis has got causality of biology wrong. The DNA on its own does absolutely nothing until activated by the rest of the system. DNA is not a cause in an active sense. I think it's better described as passive database, which is used by the organism to enable it to make the proteins that it requires. And also other statements of this idea that DNA is actually passive information. It doesn't determine organism traits or characteristics. This is very interesting. You might have now learned something about alternative splicing, how it works. But did you know that after a loss of information due to harmful mutations, genetic errors, the cell is able to compensate for the loss of information by programming the alternative splicing machinery to function in a more efficient and complex way? That's incredible. So genetic degradation is a biological fact. It happens all over the world in all kinds of organisms. But the cell still has a function and functionality to compensate for the loss of information. So this is astonishing for me, incredible. And here are a few examples of how organisms are using alternative splicing mechanism. For example, the moth called Macrosilix maya, you can see that it has a great ability to paint pictures of flies on its wings. And seems that those flies are eating something birds poop, yes. So it's a lot to do with the camouflage. And on the left side, on top of, there's a small butterfly called Goniurellia tridens. How is it pronounced in Latin, maybe? And other examples. These all are based on alternative splicing. Nothing to do with random mutations and selection. Also, these so-called adaptive radiation, very rapid changes within these cichlids in the lakes of East African lakes. These are also based on alternative splicing. No changes in DNA are needed. Scientists may have found some changes in DNA, but those changes, they don't explain these rapid changes within those fish. Okay, let's get deeper. Do we have time? Some. Did you know that every genome of every organism is very, very compressed and packed information? It is done by using nucleosomes and histones, which are like ring or circle-shaped structures that DNA is coiled around. We can see a couple of pictures that help us understand how this compression mechanism works. So when we open up the chromosome structure, we see a chromatin structure, and again we see chromatin fiber, and we can see those circle-shaped structures called nucleosomes. And again, if we open that more, then we can see DNA strands. So here's another picture of this structure. And scientists use terms such as heterochromatin, which means closed chromatin, That means that it's some kind of suppressed information. It's hidden. The cell doesn't want to read it. It's silenced information. And when the DNA is transcribed, the structure needs to be opened. So that kind of structure is called euchromatin, and it's loosely packed, and that might open the structure, and transcription can happen. And please focus on those small tails, on those sector-shaped structures called histones. Those tails have a very, very important purpose for gene expression and many, many other mechanisms and information handling systems. Those epigenetic marks on those histone tails, these are known to be at least 20 different types today. And they have many control and regulatory functions, such as they regulate alternative splicing, they determine how the methyl groups are located on top of DNA, they affect post-transcriptional modification and also post-translational modifications. So several important jobs and function tasks. Histone epigenetic marks function like a biological memory registry database. They synchronize and coordinate multiple layers of information to work together and in a timely manner. And we can think that and ask, are those histone marks inherited? Very, very interesting mechanism, how these are inherited. So here we have a larger picture of histones. The whole structure is a nucleosome, and these 4 different parts are called histones. And from every histone, in every histone there is a histone tail that may carry even hundreds of different kind of epigenetic marks. Those are mostly amino acids, consisted of from 4 to 9 atoms. So we are at atom level right now. So it's very difficult to analyze and study and make pictures of these, because the technology doesn't allow to go so deep on the atom level, at the atom level. But what is interesting is that these histone marks, and especially their combinations, they affect in many ways how the cell uses DNA and how it modifies RNA molecules, etc. They have very, very many tasks and functionalities. And scientists have to, they try to solve the histone code. It is called histone code. Today they have solved about 1%, perhaps not even that. The code is so complex, it's very complex. And because histone markers, it's information about information that might be information of another layer of information. So it's information about information about information about information, which means metadata of metadata. Evolution, of course. Sounds very designed, yes. So today, okay, there are 12 different marker types, but I should update the number. It's almost 20 or even more than 20 different marker types today. Very complex code, heritable information, and this is what epigenetic memory is based on. And Mr. Darwin also had some kind of idea of heritable memory. Yes, gemmules, yes. And he talked about the fitness, and it has something to do with memory. But today we know that this memory is based on epigenetic markers that are heritable. Then we can have a closer look at RNA signaling molecules. There are different types of these RNA molecules, and we should know that there are more than 200 different epigenetic marker types known to attach to RNA molecules. At DNA level, we only know about 4 or 5 different information marker types that can attach to DNA. But at the RNA level, there are more than 200 different marker types. And this is also some kind of biological code. It's a programming code, and scientists try to solve it. But again, the mission, yes. Yes, they have a lot of tasks, depending on their job. But if they are signaling molecules, they transmit signals from the, for example, environment and environmental factors. And by carrying different epigenetic markers, they carry the signal into the cell, and further they can bring the information to those histones and histone tails. And of course, they affect many biological pathways. Yes, but a lot of functionality. They also regulate cellular differentiation programming. They regulate tissue type, organ function, and even body plan. So the outcome, it doesn't come from the DNA. It comes from epigenetic regulation. And this slide is very important to understand. So let's have a look at this slide and stay with this a little more time. The direction and transfer chain of biological information when we're talking about adaptation of organisms. So there are some kind of environmental factors, and then the organism senses and receptors. They monitor environmental factors, and further receptors or the brain transmits this information to cells through signaling pathways using various signaling molecules. And again, the cell's epigenome responds to these signals, and the cell's epigenetic program changes. And after that, the function of the cell, tissues, and organs adapt to changes in the environment, and the changes are inherited by offspring. So any change is based on complex mechanisms, nothing to do with random mutations or selection. This slide might better answer to a question. For example, long non-coding RNAs, they have these kind of extremely complex and important tasks. They can regulate and control cell differentiation programming, direct and regulate formation of healthy tissue type, mediate and control organs to work correctly, regulate and control body plan, etc. And when evolution believers often want to compare human and chimp genomes and DNA. Okay, today we know that even at the DNA level, the similarities are about 86%. But at the long non-coding RNA level, that are very important in terms of cell differentiation, so on, the similarities are about 10% only. That's mainly because of we humans have more than 9 times more of these complex long non-coding RNAs than chimps have. And we should know that these molecules, they don't tolerate mutations. even one single mistake in one single RNA molecule might lead to serious diseases such as cancer. There are several signaling pathways in our cells, and I will not stay with this slide a long time because we don't have time to do that. You can find this information by reading the book I've authored 2-3 years ago, and it's translated into English this year. You can find this from Finnish Creation Society, luominen.fi, and the other book I've written together with Dr. Pekka Reinikainen in Finland, Game Over Darwin. This book is based on a basketball game between a platypus and Darwin. And I can say that platypus wins the game by points 251 to 0. Yes. Can I find the picture? Here, summing up, you can see something. Yeah. You can find this in Amazon. Game Over Darwin, €31. Okay, epigenetic information is dynamic and reversible because there are specific enzymes that are responsible for writing information, reading information, and even erasing information. So there are very specific mechanisms doing all that, so that the adaptation is a very dynamic process. A rapid response is possible due to an efficiently functioning information layer called the epigenome, which is kind of response some kind of buffer that handles the information between the cell and the environment. It's called the epigenome. And it is based on epigenetic readers, writers, and erasers. And here's a picture illustrating how they're working. So there are writers writing those markers to those histone tails. There are erasers taking them out, and readers that can read what kind of markers are there on those tails. And we know that you may have heard of epigenetic switches, but actually they work like dimmer switches, like knobs. And this mechanism is based on three-dimensional shape of the chromatin. And if we add to that dimensionality, we add time aspect, then we will talk about four-dimensional genome. So it's called, okay, I'll come to that later. Epigenetic memory, I just talked about it a few minutes ago. There are different types of epigenetic memory in cells, and we can distinguish between them on how stable the epigenetic modifications are in terms of inheritance. There are different types of epigenetic memory. Some of them are more stable than other types of memory. But the most fascinating mechanism is extracellular vesicles that the cell uses for passing this epigenetic memory not only to neighboring cells, but the cells from ancestors to the offspring. This occurs during the physiology of reproduction. And in addition to the inheritance of DNA, non-coding RNA molecules transmit epigenetic information to the tails of histones in embryonic cells, which direct the setting of epigenetic profiles in the embryo and fetus. This is an incredible mechanism, and this is something we should understand. We know that the methylation profiles, those CH roots, they are usually weakly inherited, and they mostly are erased during the first stages of embryonic development. But there is much stronger and stable mechanisms to transmit epigenetic information across and over generations. So it means that traits and characteristics, adaptational states and so on, they are stored in the histone database. And again, non-coding RNAs, which are encapsulated in the small nano-sized bubbles called extracellular vesicles, they transmit this kind of complex information to the next generation in the physiology of reproduction. Before we look at that mechanism more accurate level, we should know that the cell has also a function and ability to do intentional changes in DNA. So the theory of random mutations, now we know it's really outdated, it doesn't work anymore. If we talk about random mutation, then we can talk only about genetic mistakes and errors. But this is fascinating. The cell can make intentional changes in DNA, and of course RNA too, which can and do lead changes in phenotypes. A very recent discovery is the mechanism by which the cell can, by purpose, modify both RNA and DNA sequences. And it uses specific enzymes for doing this job. It uses ADAR and ApoBec and A3G enzymes. And when you read studies of this mechanism, they are quite new, and scientists are almost shocked after realizing that the cell has this kind of abilities. You can find articles by using keywords such as mutations are not random. Yeah. Yes, the environment factors might trigger the cell to make those intentional changes, and it's lots about adaptation, how the organism has to adapt to different environmental situations and factors. Yes. Speciation and species. Yes. Both, yes, yes. And at individual level, during our own lifetime, our cells can make intentional modifications that might affect protein production. Not direct examples right now, because this is also quite new for me, and I haven't found living examples yet, but I'm studying. Yeah. So, speciation is a man-made word, because today we know that most of adaptational events and so-called speciation events are based on both alternative splicing and also RNA editing. And the third one which is an important source for phenotypic variation, is so-called genetic mistakes, errors. Because at some rate, the cell has to rearrange DNA after the loss of information. The cell has to rearrange DNA in order to have correct and enough those GC pairs compared to AT pairs. There has to be enough GC pairs in the genome in order for the cell to have enough information to construct RNA molecules and proteins, etc. So different mechanisms behind speciation, but none of them results in any kind of evolution. No. They cause variation, but nothing to do with assumed evolution. No information increase. And RNA A to I editing is a very, very complex and astonishing mechanism. I can tell you something about that a little later. So we know that so-called speciation might happen without a single change in the DNA sequence, and it also might happen very quickly, only after a couple of generations. We have some examples of that. Mostly speciation is based on epigenetic regulation, which causes also errors in DNA, and this results in that the cell rearranges the DNA, and after DNA rearrangement procedures we can observe different phenotypes, and then we can say that, okay, speciation has happened. But actually it's about variation, and we can compare it to dog breeding. So RNA editing is a quite complex and but also fascinating mechanism, especially A to I RNA editing, because A to I refers to the modification of adenosine into inosine. Inosine is not a base found in RNA. Rather, adenosine has been modified by an enzyme from the ADAR family, transforming it into inosine. When messenger RNA containing inosine goes through the translation process during protein production, the translator, which is the ribosome, interprets inosine as guanine, which is the fundamental base in RNA. Thus, the event is equivalent to an A to G change in DNA, but this is a temporary change and also reversible change, just like other RNA editing events. So this is not the only RNA editing mechanism the cell has. In fact, it can change any RNA base to another base. But this is the most common and maybe the most studied mechanism. But it's incredible because we humans, and especially those who are behind this desk, our technicians, they know the network address translation system. You have heard about that. It's the same system. Yes. Because it's about masquerading, you know. Yes. So it appears as guanine to the translator, and we humans use this kind of similar technique in network traffic through what is known as network address translation, and it's a masquerading technique. So the cell has ability to make all these changes at RNA level, from any base to another. Some of them are less studied and some more studied. And now we are coming to the last, but maybe also the most complex mechanisms of how these epigenetic information layers are inherited. And now, in the beginning of this presentation, I asked you to remember a few words that are mentioned in the Bible that describes how God created the kinds and how the barriers and boundaries between kinds are maintained. And now we are coming to that information. Those nano-sized bubbles called extracellular vesicles, such as they are called exosomes and microvesicles, they can carry and transport, for example, non-coding RNA molecules, also proteins and snippets of DNA too, so several types of information. They can transport them between cells and even across generations. How is it done? Let's see. These non-coding RNAs, they influence and affect recipient cells by modifying gene expression patterns and chromatin structure. For example, sperm-derived small RNAs can carry environmental epigenetic information influencing offspring development. And the transmission of epigenetic signals through extracellular vesicles provides a non-DNA based mechanism for adaptation and inheritance. And here's an interesting statement. It's picked up from a study made in 2024. It's pretty new. So these extracellular vesicles actively modify DNA, RNA, and histone profiles in recipient cells by delivering RNA and protein cargoes for downstream epigenetic enzyme regulation. So here's a picture of a vesicle, microvesicle. It's a nano-sized bubble. It has its own membrane. And we can see structures called tetraspanins and integrins, also lectins, ligands, receptors. What are these? What is this purpose? They act like keys, and they only fit to certain locks. So the keys has to fit with a certain lock in order to release its cargo. So the cargo of the extracellular vesicle can enter the recipient cell only if the key lock pair is compatible. And this is it's not only one, but maybe the last and strongest barrier and boundary mechanism that ensures that kinds are separated and distinct from each other. Because a dog's vesicles carried in its semen fluid, its vesicles are not compatible with cat's cells. So the vesicles, the keys of the vesicle of dog, they don't match. They are not compatible with the cat's cells, which has its own locks. So they have to be compatible in order for the cargo to be released for the recipient cell. So we now have a very complex and incredible molecular level key lock mechanism behind biblical kinds. And now we have a better understanding of why barriers between different kinds are maintained and how they are maintained. There are, of course, other mechanisms, but this is one of the most fascinating mechanisms. So, key lock mechanisms are incompatible across different kinds. And interesting is also that when the cargo is released from the vesicle, it doesn't mean that it releases all of its cargo, because it's key dependent. If the cell and the vesicle uses a certain key to a certain lock, it might release only certain RNA molecules, or certain proteins, or certain DNA snippets. So it's filtered information. Incredible mechanism. Unfortunately, epigenetic regulation results in genetic degradation. You can find information by using keywords, hotspot mutations, and it's about it's associated with the methylation of cytosine mostly. But because of the lack of time, I will not handle this question long, so let's go this very quickly right now. But the main result is that so-called GC content, that means GC pairs in DNA, it gradually shifts to AT content, and this leads to problems in the cell. The problems are a loss of genetic diversity, the cell's ability to produce a variety of proteins diminishes, and the immune system weakens. Eventually, the organism is under the threat of extinction. But there are certain profiles called CPG islands. It means that there are several CG pairs in a row, side by side, even thousands of them. The cell uses these kind of CPG islands for gene or regulation or transcription regulation. Means that where transcription starts and where it stops, the intensity and duration and strength of transcription. But due to one specific mechanism, when a cytosine base is methylated, it easily converts to thymine. And this results that to a GC pair easily turn to AT pair, especially if the cell is replicated. And we should know that the cell has no mechanism to rebuild these kind of CPG islands. So it means that these islands, they only collapse. There is no known mechanism by which the cell could rebuild CPG islands. Though this argument, a very strong argument against the theory of evolution. I have challenged several artificial intelligence applications, such as ChatGPT or Gemini or other, and they have admitted that yes, there is no mechanism to build new CPG islands. So evolution has no mechanism. Genetic decay is an inevitable phenomenon. The cell tries to balance and correct this situation by copying more GC pairs by using, for example, transposable elements, but it cannot do it in such a way that it could repair the situation. It means that genetic degradation is an inevitable event in all kinds of organisms in the world. You can find by studying statistics in thisgenet.com, you can find that there are more than 2 million genetics. They are called gene disease associations in the human genome worldwide at population level. More than 2 million genetic mistakes. However, the human chromosome number is not reduced because of some certain long non-coding RNA molecules and proteins called proteins. We humans, we don't speciate because the Bible says we are created in God's image. Yes, we don't speciate. And today we know the mechanism why we don't speciate. A few words about mutational load. This is something that every serious scientist and researcher knows. They know that if much more than 3% of genome is functional, evolution becomes a destructive process. And the percentage might change. Someone has said that the percent is 10% or something. Whatever, today we know that more than 90% of the human genome is to be transcribed. And for the rest of the genome, scientists have found several regulatory functions and functionalities. So the fact is that mutational load remains, but there is no buffering junk DNA layer. So what are our conclusions? Information loss is easily observed by analyzing, for example, number of chromosomes within diploid organisms such as mammals. This example is within canids. We can see wolves still have 78 chromosomes, but after 7 speciation steps, red foxes only have 34 chromosomes. A very rapid loss of information. And here are the other examples: horses, muntjacs, mice, giraffes, and so on. So genetic entropy is an inevitable phenomenon. The best example is the famous Lenski's experiment. Did you know that after about 90,000 generations of bacteria, they have lost about 3% of their DNA due to genetic errors? That experiment started in 1988, but after 90,000 generations only. So we should also know that deletions are 4 times more common than any type of insertions. This is also a very strong argument against the theory of evolution. So, we are now close to make the conclusions. Epigenetic regulations or a loss of information. This is what any change in organisms is based on. Nothing to do with assumed evolution or natural selection. So our conclusions are that true science, serious science, supports the biblical creation account. And this is what the change is based on. organismal variation within created kinds occurs epigenetically through the switching of epigenetic toggles and sliders and knobs, which are based on epigenetic readers, writers, and erasers, and so on. Random mutations are not the cause of the organismal variation or adaptation. And these changes, they happen very rapidly, very fast. So we can conclude that any change in organisms is based on pre-existing biological information or a loss of information that results in a rearrangement of DNA, rearrangements of information. So there is no mechanism for evolution. All organisms are genetically deteriorating. We can observe a rapid decline in chromosome numbers, especially in diploid organisms. And as I said, there are more than 2 million genetic errors in the human genome. However, no human speciation. The number of fully random or fully beneficial mutations in human genome is 0. There are no beneficial mutations in the human genome. And we can ask, why would one of the most important questions, the question of the origin of life and humanity, be written metaphorically? So based on scientific observations, the biblical creation account is based on true events. Creation is mentioned over 200 times in the Bible, evolution never. The truth can be found in the Bible. So at last I want to share these verses with you. But ask now the beasts, and they shall teach thee, and the fowls of the air, and they shall tell thee. Or speak to the earth, and it shall teach thee, and the fishes of the sea shall declare unto thee. Who knoweth not in all these that the hand of the Lord hath wrought this, in whose hand is the soul of every living thing, and the breath of all mankind? So thank you very much. Vielen Dank. Gottes Segen. Yeah, 5 minutes for questions. Yes.
Hat jemand Fragen? Wir haben noch ein paar Minuten Zeit, wenn es noch Fragen gibt.
My question was, I'm not a biologist, but when you, so the sperm and the egg, when they come together, is that, because I thought the eggs are made at a very early stage, they don't change. So all the change would come through the sperm into this newborn?
Yeah, mostly the sperm cell carries the DNA, of course, chromosomes, but without those extracellular vesicles and the other information that is carried in the semen fluid, the embryo doesn't start to develop. The fertilization might happen, but it stops. The development stops if there is no information carried by those extracellular vesicles. They are necessary. But the egg, it has different mechanisms, but it also has several extracellular vesicles, but not in fluid. They are in the egg, yes.
The work of epigenetic genomes is driven by changes as well as by regular work in the body, or?
You mean changes?
Yeah. that the epigenetic genomes starts to work is driven by external things,
Yes.
and it is also driven by internal regular things.
Absolutely, yes.
Yeah.
There are some scientists who have studied, for example, how our thinking and even attitudes affect our epigenomes. And they have found several evidence that it's true, yes, it is happening. Even our thinking and attitudes affect epigenomes. True, yes.
Thanks first for your very interesting lecture. My first question would be, are the slides available somehow? Because they're very fine. Second, regarding the CPG islands you told about, is it likely to say or to assume that this repair mechanism is stopped since the fall? Mm-hmm. And the third question would be, is there any idea for applications in the healthcare sector or in the medicine area?
Yeah, first question, you are correct. After the fall of sin, that's my own opinion, but because the certain DNA repair mechanism is missing. So I think that after the fall of sin, God in some way modified the repair mechanism systems and took away one of the most important repair mechanisms. So its genome started to deteriorate. But the other question was regarding medicine, medication? Applications. Yeah. Yeah. Especially medical sciences are very interested in epigenetic studies and regulation and all kind of this stuff. Because today they more and more understand and know how these systems work, and they start to focus on the epigenetic regulation and histone marks and alternative splicing and non-coding RNAs. They don't focus on the DNA sections, not so much anymore. That's outdated, and they doesn't provide the information and the reasons for. But of course, genetic diseases is an interesting topic, and today we heard a presentation. Mr. Ola Höscher talked about SNPs, single nucleotide. Polymorphism, yes. There are small changes at nucleotide base level, and they are associated with genetic diseases very often. But yes, medical sciences are now focused on epigenetics too. Yes. Yes. Yeah. You are talking about when we're newborn. Yes. 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 True. Yes. True.
And I heard from a couple they had 2 children and then they became born again and had 2 others and told me the new one are different from the old.
Epigenetic memories is inheritable. Also, negative things such as addictions, they also are inherited. And we remember, after we have read our Bibles, that God, what it said, a 4th generation. You can remember that verse? And it's about epigenetics. Curse even to the 4th generation. But we also have heard about how our sins are washed away after we have accepted Jesus in our lives. So it's also about epigenetics, because when epigenome works correctly, then it means that it has been some kind of re-established or flushed or washed. And we can also affect our epigenome by eating healthy food, by sleeping, by having good relationship with our communities, with other people, by having physical exercise and so on. It makes good to our epigenomes. Yes. Okay.
Thank you.
Thank you.
Ja, wir sind eingeladen direkt in die nächste Pause. Draußen gibt es Kaffee, gerne auch einen Kuchen oder auch was Herzhaftes mit dabei. Und dann dürft ihr euch auch direkt in den Plan schauen, wo die nächste Session stattfindet. Die Experimente eine Etage höher, die werden in den nächsten Stunden nochmal durchgehend laufen. Wer noch nicht da war, nutzt gerne die Chance und geht auch dahin. Dann so, ja, die Pause war für den einen oder anderen sehr kurz. Trotzdem wollen wir jetzt mit dem nächsten Vortrag starten. Und bevor wir das Wort übergeben, würde ich gerade beten und dann können wir starten. Hes, ich danke dir dafür, dass wir diesen Tag erleben dürfen und so viel Input sammeln können. Hilf es uns zu verarbeiten, aber auch jetzt einfach offene Ohren für diese neuen Informationen zu haben. Und dass es einfach wieder ein Vortrag sein kann, der uns bereichert. Amen.
Welcome to this session. I will speak English the whole time, but I have my slides in German to make things a bit clearer for the German listeners, but I will then translate what the slides are saying. So this is about ultimate engineering in biology and also answering the bad design arguments. This is really relevant, especially in the United Kingdom and in the United States. Maybe it'll come a little bit later in mainland Europe. There are many books coming out claiming, for example, the human body has bad design. The books are saying we have a bad design of the eye, bad design of the back, bad design of the foot, bad design of the wrist, almost from head to toe, we are a bad design. There's two reasons for this. One reason is evolution predicts bad design. I agree with these authors, evolution predicts bad design. But of course, the problem for those authors is we are discovering brilliant design, not bad design. So I'm just completing a book, will be published in America, and the book is arguing it's not bad design, it's ultimate engineering at the limit of what is possible. So this is what I will explain in this book, but in this presentation. But in my book, I address these authors: Nathan Lentz, Abby Hafer, Richard Dawkins, Jerry Coyne, Alice Roberts. I show how they have false science, and I will explain this in the talk today. I should also add that most of these authors are fanatical atheists, and they hate God. This is actually their main motivation for their books. So let's get into it. Two world views, zwei Weltanschauung. There is the biblical world view on the left. The biblical world view is perfection, as the theme of this conference, perfection in creation. So according to the biblical worldview, the design of the human body should be better than human technology. That's the prediction of the Bible. But what is the prediction of evolution? The prediction of evolution is biology should be worse than human technology, because evolution is very clumsy, it's step by step. Human designers are not limited that way. So evolution predicts biology worse than human technology. The Bible predicts biology better than human technology. It's a very interesting contrast of worldviews. So why do I say that? Well, in the Bible, it says God's way is perfect. Many verses in the Bible says God does things in a perfect way, perfect judgment, perfect providence, perfect salvation, perfect design. God is capable of perfect design. He has perfect knowledge of physics, of chemistry. But I would go further and say God can only do perfect design. It's his character. He can't do imperfect design. He can only do perfection of design. Of course, there is an objection. Well, we live in an imperfect world. Because of the fall of Adam and Eve, we now have genetic decay. But if you understand what the genetic decay is, if you understand that, you can then understand what is the perfect design. Because you can say the same about a motor car. BMW motorcars are a brilliant design. Ich liebe BMW motorcars. Achte BMW. But a BMW car gets old, it gets rusty, it can break down, and if there was some bad information, it wouldn't work. But this is different from the design as it was intended. And so it is with the human being. We get old, things go wrong, but the original design was ultimate engineering. So we must not get confused between the original design and death and disease. Many non-Christians get really confused between design and disease. We mustn't confuse the two things. This is an important slide. This is a senior biologist from Duke University. Now, what he is saying here is that the evolution process is very restricted, very constrained by step-by-step change. And therefore, evolution cannot produce very inventive designs. It's really restricted. And then he makes a really interesting admission. We do not publicize this restriction. We don't tell the students about this restriction. And I can give you other quotations where the evolutionist says, we will keep quiet about this restriction. I find one of the most frustrating things, I can speak to my colleagues in a university, other professors, say of biology, and they will say, there are many problems with evolution, many problems with abiogenesis, but they do not explain that to the students or the children in the schools. This is really frustrating. They tell the students, everything is fine, we can be confident. But when we're in the university, they say we're not confident at all, but don't tell the students. I find this the most frustrating thing. So this is an example of this admission. So these are 3 examples of books that claim bad design. One is by Jerry Coyne, another by Nathan Lentz, another by Abby Hafer. I could also add Richard Dawkins and Alice Roberts, but these are the 3 most important. In fact, the middle one, Nathan Lentz, has a lot of publicity in the United States and in the United Kingdom. And so they're claiming the eye is badly designed, the back is badly designed, the joints are badly designed. And this is, sadly, this is what's getting into schools and colleges, not the real science, but these, as I will explain, these claims. But what's interesting with these books, Jerry Coyne, he says, we expect evolution to produce imperfect design. And Nathan Lentz says, evolution is worse than an engineer, it's worse than technology. And Abby Hafer, we expect a failure grade from evolution. Now, I agree with these statements. Evolution does predict schlecht design, bad design. But the problem for them is the reality does not support their predictions. And later I will show you there's a tension between some biologists and other biologists who are disagreeing with this. It's interesting. So I have produced this graph. This is another important slide. And this will be in my book. So we have complexity on the left, rising complexity. Then we have time along here. According to evolution, things started at ground zero. And evolution is gradually increasing complexity of organisms. It will never catch up with human technology because it's restricted. So this is what evolution predicts. But intelligent design, or the biblical worldview, predicts things started from ultimate engineering and is now in a process of decay, but still way above human technology. And of course, what we observe is exactly this decay from a perfect state. It's really encouraging. What we observe in biology exactly supports the Bible. Science supports the Bible. We are clearly seeing this genetic decay. We've had talks, in fact, the last talk in here was describing this genetic decay, and other talks have described that. And John Samford and Michael Behe and others have talked about devolution, genetic decay. So this is well known amongst ID people. And every year we see creatures becoming extinct. We are observing genetic decay in the human being. But if you can identify what is the genetic decay, then you can observe ultimate engineering. And in the whole body, we see ultimate engineering at the limit of what is possible. So briefly, I will go through these 10 areas. The cardiovascular system, the eye, the nervous system, the throat, the appendix, birth, back, foot, knee, hand. There's two reasons why I have this list. In these 10 areas, those previous books by the atheists, they claim these are badly designed. They claim bad design. But also, I'm interested in these areas. My speciality is biomechanics. The biomechanics not just of joints, but the biomechanics of any muscle. And muscles are used in all of this, in the heart, in the eye. The nervous system is integrated with muscles. So as a biomechanics engineer, a researcher, I'm interested in all of these, but particularly in the bottom three. I have a lot of publications in the bottom three. I have about 200 scientific publications on the science of design in engineering and in biology. And I've been an editor of one bioengineering journal. So we go through these quite quickly. The first one is a good example. So I'm going to give you a quotation from Professor Sean Harding. She is a world expert in the heart. And she's going to say something which I really agree with. This is really true. Nothing shows more clearly the perfect design of the heart than our own failed attempts of engineers to copy the heart. So this is a world expert who has studied the heart for 40 years, and after 40 years, her conclusion is the heart is perfect. Exactly the topic of our conference. The heart is perfect. Engineers can't copy it. But Richard Dawkins says the heart is a bad design. He's never researched the heart. So we have a world-leading cardiologist. After 40 years, their conclusion is the heart is perfect. But Richard Dawkins, who's never looked at the heart, he says the heart's bad. Why does he say the heart's bad? Well, two reasons. First of all, he doesn't read what the scientists are concluding. Secondly, evolution predicts the heart is a bad design. And because evolution predicts the heart should be a bad design, he says it's a bad design, not based on science, but based on his worldview, Weltanschauung. And the worst thing, what is the worst thing? Students are told this. They're not told this one, the true science. They're told this one. So my first example is quite extreme. It's quite an extreme example. Observational science just tells you the heart is perfect. But evolution predicts it's bad. And so this is the first example. The second example, the eye. I have just seen on the second floor the creation exhibition by Peter Schunke and others. And I really recommend you must see this exhibition. When I was going round with the headphones, they have an English option. I went round with the headphones and it said the eye is at the limit of performance because the light detecting cells are so small they can measure 1 photon of light. When you go through the human body, you keep finding everything is at the limit of performance, down to the molecular level, whether it's the nervous system, DNA, it's at the limit of what is possible. Did you know that 1 gram of DNA has the same similar storage capacity as 1 million flash drives, also of 1 gram. So you need 1 million times more flash drives of the same weight as 1 gram of DNA. DNA is at the limit of what is physically possible. So the eye is at the limit of what is possible. I'm interested mostly in the biomechanics of the eye. It's one of the most used, or the most used muscles in the whole body. The jaw is one, and the eyes are the others for the most used muscles in the body. And some special design features. But this is what Dawkins says about the eye. Das Auge ist nicht nur schlechtes Design, es ist das Design eines kompletten Idioten. It's not just bad design, it's the design of a complete idiot. So why does Richard Dawkins say this? Well, he claims that because the light has to go through the retina to get to the cells at the back, it must be degraded as it goes through the retina. But as explained in the exhibition on the second floor, it explains scientists have discovered these Muller cells, which guide the light through the retina. And not only does it mean that it's not degraded, but it actually does signal conditioning. It deals with reflected light. So what Richard Dawkins thinks is a terrible design is actually a strength of the design. And it's a classic example of an evolutionist, because of evolutionary worldview, they start with the assumption, this looks very complicated, it must be a bad design. Not in humility thinking, I don't understand this, it might be a brilliant design. That's how a Christian would look at it. They say, I don't understand it, so it must be terrible. So I'll just assume it's terrible, and I'll teach the students it's terrible. So the books I showed you earlier by Abby Hafer, Nathan Lentz, they still have this mistake. And those earlier books were published after this research, not just in 2010, but in 2015. This was published in Scientific American. In 2015, it became wide knowledge. And the books by Nathan Lentz and Abby Hafer were published after 2015. They're still publishing this claim, which is being proven to be completely wrong. And students are being taught this false, bad design argument. So I'll be explaining this one in my book as well. Number 3, wunderbare nervous system, amazing nervous system. I can appreciate the nervous system myself because I designed most of the wiring on the Envisat satellite, the world's largest Earth observation satellite. It would fill this room, this satellite. And I designed the wiring for the solar array, which was the size of a badminton court. There were 400 wires. The distance to the end of the solar array was more than 20 meters. And if you add it all up, it comes to nearly 10 kilometers of wiring I had to do myself. I was in charge of the design and the wiring for the solar array. And I had to get the wires from one end of the spacecraft to the other end, and it requires a lot of planning. And I had to put holes through structures to get the wires to the right place, which meant those structures had to be redesigned and planned for the wire to go through. A huge amount of coordination. And what's interesting is that in the human skull, there are around 21 holes for wiring. And when I look at the human skull and I see these 21 holes for various nerves and veins, the jugular vein, the optic nerve for the eyes, the nerves for the other parts, when I look at these holes in the skull, I think that's exactly what I had to do on a spacecraft. I had to have these holes and to put the wires through. It needs immense planning. But in the case of the human nervous system, there is 150,000 kilometers of wiring. It just dwarfs what a spacecraft has. In a spacecraft, you have to do so much planning. So this is incomprehensible. It reaches every corner of the human body, the hierarchical distribution of wires. Engineers are in awe of the human nervous system. Often it's the wiring which is the most complicated, the most integrated part of a system. And a spacecraft is the most advanced technology, and yet the human body is just on another level. So why am I saying this? An engineer would not dare to pick fault with the human nervous system. How could you try to pick a fault with the human nervous system? You just cannot do that. It's so beyond anything engineers can do. And yet, Richard Dawkins, he says, is a bad design. He's never designed a wiring system. He's never designed anything. So he says, you know, the innards of an animal is just a complete mess. And this is a really bad design. So you may have heard about the laryngeal nerve, the recurrent laryngeal nerve. That's this one here. The superior one is the one in the red at the top. The laryngeal one is this one. So Richard Dawkins says this one that comes down to the heart, he looks at it and says, that looks wrong to me because it's a loop. And therefore he publishes it, and it's been on television, and millions of students think, this is a bad design because it comes down. Now, I'm different to Richard Dawkins because I have done wiring, and I teach it, and I practice it, and I've done it on a spacecraft. When I first saw that, what do you think my reaction was? That is exactly what an engineer does. That is exactly what I would expect. When an engineer does wiring, you often have loops. Loops in your car, your house. You have loops in the wiring. That is what engineers do. That's exactly what you'd expect. But of course, Richard Dawkins, he didn't ask an engineer. Evolution predicts bad design, so I just pick faults with that. This is part of a spacecraft I designed for a European satellite. I designed the spacecraft. Now, you can see two loops. In fact, those loops are very similar in size to the laryngeal nerve. On here, can you see a blue wire and a red wire? They are sensors for a hinge. One is a temperature sensor, and one is another kind of sensor. Those wires are so small, they cannot go on their own. So they piggyback on a bigger bundle of wires. Now, why am I saying that? Sometimes in wiring, it's best to do a little detour so you can pick up another smaller wire, which will then be strengthened by the bigger bundle, and then you do your routing. Sometimes you have a loop because when the technician is assembling, you can't physically assemble it unless you have some slack in the system. In here, you also have a loop because the hinge is moving and you need some flexibility, and our necks move. And also for redundancy as well, you have a backup. But okay, so if we go back to this one, notice there are intermediate connections to the esophagus and to the trachea. So these are like my sensor connections. So this loop is not only going to the larynx, it is also carrying other wires to other parts of the body. And it uses the laryngeal nerve as a strengthening backup. This is exactly what I do on my spacecraft. It also has redundancy. There are examples of soldiers being shot in the neck, but they can still speak because you have two pathways to the larynx from two different directions. If you only had the top one, you could lose your voice if you lost one nerve. Some researchers are wondering if you get a better control of the larynx by having two different pathways to the larynx, and that might be the case. But what I'm illustrating is that wiring is very, very complicated. And for an evolutionist to pick fault and say, oh, this just looks wrong to me, Richard Dawkins hasn't explained what any disadvantage of having this loop. There's no disadvantage. The extra mass is virtually nothing. When a baby grows in the womb, different parts of the baby are growing at different rates. For example, the intestines grow much faster than the rest of the baby in the womb. And so different parts of the baby are growing at different rates. And so it's inevitable that you need some loops in the nervous system for the body to grow so that you're not pulling too hard. And scientists have an incomplete understanding of how the baby grows in the womb. So it's a very, very complicated situation. But as someone who designs wiring, this just looks completely normal to me. But Richard Dawkins, who's never done any wiring, he just looks, evolution predicts bad design. And this is taught to the students. In each case, the wrong science is taught to the students. Okay, so then we come to the human throat. It's an amazing design, the human throat, because it does 3 functions. It does breathing, it does eating, and it does speaking. And in fact, not just speaking, but singing. You can sing with your mouth, you can talk with your mouth, you can eat with your mouth, and you can breathe with your mouth. So it's multifunctioning. Last year, I wrote a scientific paper, which I'll show a little bit later, called Multifunctioning in Animals. And I mentioned the mouth as a multifunctioning system. And I said, if robotics engineers want to make progress, they need to copy multifunctioning in biology. And I mentioned the mouth, although I focused on joints, multifunctioning in joints. So engineers are in awe of the human mouth, the wonderful multifunctioning system. This is one of the secrets of perfection in biology. In particular, the human voice, singing, is an incredible design. If you're interested, you can look at this paper at the bottom. A surgeon was asked to write a leading review paper, and he said the human voice is a masterpiece of design. One of the things he explained is that in the case of a musical instrument like clarinet or another type of wind instrument, an oboe or a flute, it has lots of different mechanisms for different pitch and sound. But he said the human voice only has one pair of vocal folds, just one. But instruments have lots of mechanisms. And he said that one pair of vocal folds can produce more music than these other instruments. It's an amazing design. Engineers are in awe of the human voice. But Abby Hafer, this evolutionist, she says the human throat is a really bad design. It's a terrible design. Because if you're not careful, you can choke. Well, there are lots of things. If you're not careful, then you can have all kinds of problems. And this is getting into medical books and student books. Students are told this. Students are not, they're not told the findings of a top surgeon working on the throat. They are told, Abby Hafer, who's never researched the throat, saying it's a terrible design. So our students, over and over again, the same will happen with the German students, and it's going to get worse. So Abby Hafer, well, you have to give her a little bit of credit, because unlike the other cases, she says she thinks she does know a better design. She says we should be like a whale. We should have a hole in the back of our neck, and we should speak through our mouth, and we should eat through our mouth, and we should breathe through the back of our neck. We should have 2 pairs of lips, 2 tongues, 2 mouths, one at the back and one at the front, with a neck probably this wide. That's what, you know. So she claims to be a better designer. One of the things I'm explaining in my book is, to be an engineer, you need to study for 5 years. And when you graduate after 5 years, you're told you're not ready to design. You're not ready to design. You have to go under someone else. And maybe after another 5 years, you can begin to design. And maybe after 30 years, you can review a design and be a judge of design. And these people, if they were in my first year class, they would fail the first year student exam on design. And yet they are teaching biologists what they think is good design and bad design. Then we have the appendix. Abby Hafer says, you know, this is a terrible design, works best in rabbits, not in humans. But she's unaware of research which shows that humans are better off with their appendix. It helps your immune system. Of course, there are some parts of the human body you can survive without an appendix. You can survive with 1 arm or no arms. But you're better off not to lose your arms or your appendix. But evolution predicts bad design. So, yeah, I think this is another case of bad design. You may not know this, but when a woman gives birth to a baby, one of the most interesting things is the biomechanics of the pelvic floor. I have seen 5 births. My wife's had 5 babies. And I have to confess, I was really interested in the biomechanics during birth, because the pelvic floor muscles, they expand more than any other muscle in the body during birth, and very fast. They expand over 300%, 320%. And to do that, they need hormones. It's the most incredible process. So your pelvic floor muscles go from the top one to the bottom one, an enormous change. And it requires a lot of hormones, and also the baby's skull goes down. It's a miracle of design, and you might have heard the cardiovascular system has to reconfigure. There's a flap in the heart that closes, and the plumbing is different. It's the most miraculous thing. But Abby Hafer says, you know, this is a terrible design, human birth. And again, she thinks she knows a better design. She thinks that humans should be like a kangaroo and have a pouch. She thinks this would be a better design. But she seems to be unaware that the human being has a lot of development in the womb. They can't actually be born very early. But so you have to give Abby Hafer credit. She is trying to say, well, this is what I would do for a better design. But again, this is what students are taught today. Then we come to the human back. Is this a good design or a bad design? Evolution predicts a bad design. I should add at this point, for the next few examples, if you read books on evolution, they say the human back and our joints in our legs are really bad design because we assume we have come from apes who walk on all fours, and there was not enough time to evolve to be upright. And because there was not enough time, it's impossible that we have a good design of our back, our feet, our knees, our hip. There must be a bad design because it was so fast to go from a quadruped to a biped. So the books say a bad design. Well, I point out in my book, engineers are spending millions of euros copying the human back because it's such a brilliant design. Engineers have studied the human back and they say, this is fantastic. We need to make a bridge like the human back because the joints are so efficient. So this is science. This is reality. Reality is engineers are in awe of the human back because it is both strong and flexible. It's really strong, but with all of these vertebrae and the joints, you actually have this flexibility. Of course, our modern lifestyle of sitting too much is not good for the human back. And a lot of people suffer from a bad back. But if you go back 100 years where people walked and cycled and worked in the field, it was much less common to have a back problem. We do too much sitting today. So Nathan Lentz says, the back, it's a bad design, you know. Must be a bad design because it took a long time to stand up. So now students are being told, the back's a bad design because evolution says it's a bad design. They're not told that engineers are copying the human back. They're not told the true science. They're told the fake science. The human foot. I've published, in recent years, I've published several papers on the human foot, including prosthetic designs. Some of my papers have won some prizes, and I explain that it's a brilliant design, far better than any prosthetic foot. You know, you can get a prosthetic knee joint, you can get a prosthetic hip joint, you cannot get a prosthetic foot. Why? It's too difficult to copy the human foot. It's a brilliant design. In particular, the human foot can transition between stiff for running and flexible for landing. And engineers cannot crack this problem of stiff and flexible, stiff and flexible. If you're interested, you can look up this article on the human foot, where I also answer bad design arguments. I've also published this paper, which talks about engineers should copy the human foot. But Nathan Lentz, particularly the ankle, knuckle, it's a terrible design. It's a terrible design. And he says it would be better if you just fused all the joints. And he's unaware that if you fuse the joints in the knee joint, in the ankle joint, you lose functionality. It's really well known in the scientific community. So he starts saying things that contradict really like first year level undergraduate biology or medicine. It's quite incredible. The evolution theory gives him a license to make these wild claims which just contradict basic science. And the same with the knee joints. I've published a lot of papers over the last 20 years on the human knee joint, particularly the four-bar mechanism of the knee joint. It's an incredible joint with a large range of motion, a lot of engineering in the knee joint, irreducibly complex engineering. I've had a PhD student just work on the knee joint for 3 years. And even after 3 years, we realized there's a lot more that we could do. We produced mathematical models of the 4-bar linkage system. This is an irreducibly complex 4-bar linkage mechanism produced by the cruciate ligaments, just like a car suspension system, like a double wishbone system that cannot evolve step by step. It's a brilliant design, and I can show you quotations from other researchers saying, you know, joints are an amazing design. Synovial fluid is the most incredible design. But once again, Nathan Lentz says, the knee's a bad design. It must be a bad design, because if we evolved from apes, it's got to be a bad design. He hasn't spoken with researchers. He hasn't asked engineers. He just says it must be a bad design. So we should teach students it's a bad design purely because evolution predicts a bad design. Can you see how damaging the evolution theory is to science? It's destructive to science. It's destructive to what students are being taught. Ironically, the evolutionist has said, you know, how bad if we follow intelligent design when it's evolution which is destroying science. And then the hand. And I'll just mention the wrist. I published a paper with the wrist joint as a main example of the most incredible example of multi-functioning. And I explained that engineers cannot produce a prosthetic wrist joint as small as our wrist joint. It's very small, the wrist joint. And it can move in 3 different directions. And engineers cannot produce a prosthetic wrist joint that small. They're always bulky. So if you're interested, you can look up this paper, Biomimetics 2024, how multifunctioning joints produced highly agile limbs in animals with lessons for robotics. It's a secular journal. This won a Best Paper prize. And I showed the same 8 bones in the wrist. I said it's a genius design because the same 8 bones are in the perfect place to perform 4 different functions in different planes. And I imply this is at the limit of what is possible in the wrist joint. And I said engineers need to copy this ingenious design. And it won a paper prize. Well, what do you think Nathan Lentz said about the wrist joint? He said it's just a useless pile of like a useless pile of rocks. The wrist joint, they're just useless bones. So students are taught the bones of your wrist are just useless bones. And I've just written a paper saying they are the most ingenious design, like a Rolex watch. It's just like Nathan Lentz looks at a Rolex watch and says, what a useless pile of metal that Rolex watch is. I can't understand what it is, so it must be a useless pile of metal. In my paper, Multifunctioning Joints, this is a secular paper, I wrote, this is a major challenge to evolution. This is an example of irreducible complexity. And the reviewers let me publish this paper. So this was a big encouragement. I said, this is a major challenge to evolution because you can't plan these multifunctions in one go. According to evolution, the purpose of your hand is to form a fist and punch someone in the face. That's what evolution says. And there's the paper. That's the paper. Or hold a club and hit someone on the head. Which is very different to the biblical worldview of we are designed to play musical instruments. By the way, the reason we can form a fist is so we can hold a squash racket and play that brilliant sport. I had another encouragement last year because this relates to bad design. One of the icons of evolution is the vertebrate limb. This is in the biology books around the world, the vertebrate limb. Darwin said, this is my inspiration for evolution because the bone layout is the same in humans, the whale, the bird, the leg, the frog, and so on. But what was his assumption? His assumption was bad design. He believed the whale flipper did not need a wrist, and it did not need fingers, and it didn't need the muscles in the fingers to flex. And the same for the bird. He said, I assume it's a bad design. Over the decades, I've come to realize Darwin was spectacularly wrong. The flipper is not a bad design. The whale flipper does need the fingers. It does need the wrist. And so I wrote an enormous paper, 15,000 words, 50 diagrams, explaining the hydrodynamics of how whales swim and the aerodynamics of how birds fly. Of course, Darwin didn't understand that. And I also showed papers, recent papers, that showed that whales do move their fingers in their flippers. And their muscles are fully functioning. They have muscles like we have, and they move them, and they need them. Why do they need their fingers? To fine-tune stiffness and shape of their flippers. They actually use their muscles. When a whale is decelerating, there's huge forces on their flippers, and they need their fingers to work to stop the flippers deforming. If the flippers deform too much, then the hydrodynamic drag increases because the drag coefficient is not good because of the shape of the flippers. But of course, Darwin didn't know that. And so I wrote a paper saying universal optimal design in the vertebrate limb, because Darwin said it's not universally optimal. So I thought I'll put this in the title, Universal Optimal Design, in defiance of Darwin. And in the paper, I said, this paper challenges Darwin's homology argument. And I submitted it to the Institute of Physics. And I thought they would say, no, this, you cannot contradict Darwin. And they said, yeah, we'll publish this. And we will make it a review paper, a state-of-the-art paper, review paper, which meant it was golden access. So normally you have to pay to see the paper, but it's free because it's a review paper. And they said, yeah, well, the reason they said yes is because they said engineers need to know this. Engineers need to know what is true, because engineers need to know what should we copy. And evolution was saying, don't copy that, don't copy that. But engineers want to know what is the... Engineers are very pragmatic. They want to know the truth because they want their company to be successful. So they said, yeah, we need to know this. That Darwin was wrong. This is an icon of evolution. But of course, this isn't going to make the media, because the media want to tell you what Nathan Lent says and Abby Hafer and Richard Dawkins, which is just the anti-science. So this is very near the end. This is a world-leading biologist who does world-leading research, observational science. Biologists are often awestruck by the perfection of biological systems. He's an Italian scientist with 7,500 citations. He's a world-leading biologist. He's an observational scientist who does work in the laboratory. So you have two groups of biologists. You have those who do real observational work, and they say biology has perfection. Then you have another group over here who follow evolutionary philosophy. And they say biology is a bad design, because that's what evolution predicts. So it must be bad. And they're not reading papers by people like him saying, well, actually, biology is perfection. So we have these two groups. But what is sad is that our education system is teaching this group, not this group. Students are not told this. They're not told the heart is a perfect design by the world leader in heart cardiology. They're told what the evolutionists are teaching. In Psalm 33, it says, stand, when you consider creation, we should stand in awe of God. Just like this biology, we are awestruck. Psalm 33, we should stand in awe of God. These authors, Nathan Lentz does not want people to stand in awe of God. I think he hates God. This is why he's pushing the evolutionary theory, and he wants students not to stand in awe of God. So these are the previous books, but in February you can look out for my book, Ultimate Engineering, where I take on these authors. I hope you found that encouraging. Thank you. Yeah, sorry, yes. We have time for just a few questions. Yeah, it's a good question. Birth is a special case because that was particularly mentioned. Birth is a special case, but still, you can identify some amazing design features in birth. But pain is a part of the fall, particularly with giving birth. So we have to be careful with that particular example. Thank you.
Hi, thank you so much for the interesting and encouraging
for humans. There's no advantage to go to that other kind of eye. I just give it a quick illustration. There's also people arguing our nose is not as good as a dog, and our eyes are not as our smell and our hearing is not as good as a dog. It's really interesting. In the last couple of years, there have been publications, and the scientists have said, actually, this is a mistake. Human smell and human hearing is actually better than animals like dogs. Because, for example, with our hearing, we have the best ability to differentiate between really similar sounds. And when we're speaking, we need to differentiate between very similar sounds. And similar with smell, we're very good at differentiating. Now, what an engineer will explain is that you always have a trade-off. You cannot have everything. You cannot have the strongest sense of hearing and also a sense of differentiation. So God has given us exactly what we need. We don't need to be able to have a 1 in a billion sense of smell. But what we do need is to differentiate. And with hearing, we have this superior hearing, differentiating. And it'll be the same with the eye. You can't have everything. We don't need to see like an eagle sees or an octopus sees, but we can differentiate between 10 million colors. God has given us the performance that humans need, which is different to other animals. So another aspect of design, which is quite sophisticated, is the trade-offs, that balance of different multi-function optimization and trade-off. And as someone who lectures in multi-objective optimization, I can see that with each human part, we have this perfect combination. But of course, when someone is ignorant of design and trade-offs and multi-objective optimization, it's quite hard to explain to them. But it's interesting, papers are coming out saying, actually, we made a mistake that to say humans don't have good hearing, don't have good smell, good sight. The papers are coming out saying, actually, now we're understanding they do actually have the best design. Yeah.
Do you have an idea why the inverted eye is like the best for the human? I just don't know it.
Okay, it gives a better blood supply to the cells. And given that you can have the optical fibers, the Müller cells, then with the better blood supply, you have the best design. In every case of the human body, it takes us a long time to fully understand. But once we do understand, then we say, yeah, this is the best. This is the best design.
Thank you.
Hey, I have a question because I come from the informatic field, and in the approach in Scrum or Kanban, it's like that you want to have little increments of software that you have to implement, because then you can check if you are still on track to the best design. So I don't fully understand why evolutionists say or claim that incremental steps will be a bad design, if my experience in our field of you want to have those little increments to get the best design. So I don't know how they come to that.
So I teach engineering design, and when I teach engineering design to students, I tell them, do not design by incremental change, because you will fail. If you want to design something interesting, you have to make a huge leap. If you don't do that, you'll fail. Because step-by-step change is just completely restrictive. If you have a car and you do incremental change, you're only changing the value of parameter, the width, the height, the length. What you need is an innovative new design with new parameters, new concepts, new physical effects, and that involves a gigantic leap. And the evolutionists know this. So they're correct that evolution is really, really restricted. And it's not only restricted by step change, but they recognize it's restricted in other ways. It has vestigial parts that it cannot delete. It's restricted to good enough design. It cannot design more than is necessary for survival, and it's restricted by time. There are at least 4 constraints to evolution. And it's not just me saying that, but the evolutionists agree. And this is why it's strange. I actually agree with these authors. Evolution predicts really bad design. It's just tinkering. For example, engineers use genetic algorithms. But if you look at the genetic algorithms, they are only adaptation. If you have a genetic algorithm, it cannot change one concept to another concept. If you apply it to a car, you always have a car. You never become a bicycle or an aeroplane or a space rocket. It's fixed as a car. So when we use genetic algorithms, it's proving to us exactly what we know in biology that microevolution has hard limits of change. If you do step by step, you're limited to microevolution. You cannot jump to macroevolution.
Okay, thank you.
My question is slightly related to the previous question. We know that science progressed most in regions and times when it was based on Christian worldviews. Do you see impacts on the progress in science if it's now focused on other worldviews?
Yeah, of course. I think I was implying that you have things like junk DNA. They assumed, junk DNA must be junk. If they had been more humble, thinking maybe we don't understand this, they would have saved time. They lost time by labeling junk DNA as junk DNA. And, you know, the whale flipper could have been appreciated earlier if they hadn't have just assumed it was a bad design. The same with the bird wing. They just thought that was a paddle. And also bio-inspired design. Yeah, so it does slow down. It slows science. That has a big impact on science. Yeah. Maybe one last question, and then people can ask me later because the time is gone. So one last question, and then we finish.
Yeah, you focused on the design of these individual human organs, but don't we have to see the complete picture? Because God, when he made the laws of nature, like how should the molecules behave, how should all these forces interact, strong, weak nuclear force, he already had in mind how he can create human beings, since in all humbleness we are the pinnacle of his creation, or at least the purpose of his creation. So it's not just how wonderful are these organs designed, it's how wonderful he combined the laws of nature with how to design these particular organs. So he had variables in both.
I agree with that, and this theme comes out in the wonderful exhibition on the second floor. That when you go through the exhibition, you see the whole picture from the tiniest, even the atoms and the molecules, how it all builds up eventually to our need to have a relationship with God and our emotions. So this point, thank you for the point, it comes out brilliantly in that exhibition. So I do encourage you to see that. But I think we finish there at that point. Thank you.